First of all, I just want to welcome everybody to our Juneteenth celebration. I think most of you know me. I'm Mike Friedlander, the Virginia Tech Vice President for Health Sciences and Director of the Fralin Biomedical Research Institute. The Institute is really delighted to sponsor this important event, and we're pleased to host all of you here in the auditorium and those joining us online as well. Is the online on, Brad? Good, okay. Although Juneteenth is on Sunday, June 19th, with the official federal holiday this year occurring on Monday, June 20th, we thought it was important to have this program today in close temporal proximity to the holiday to allow people to also take time on the actual holiday to reflect on its significance and to recommit to addressing injustice and racism in our personal and our professional lives. Juneteenth commemorates the anniversary of Black Independence Day, when freedom for enslaved people was proclaimed 157 years ago in Texas, the last state of the Confederacy with institutionalized slavery. 2.5 years, by the way, after the Emancipation Proclamation. However, only 1 year ago, President Biden signed the Juneteenth National Independence Day Act into law, making it a federal holiday. All great journeys of reconciliation and justice begin with stating the truth. And justice delayed is justice denied, as has been said by many scholars over the years. But we're on a path, I think, particularly in the last couple of years in this country, to come to grips with a history of injustice. And in our little world in academia and science and medicine, we have our own way of reaching out to try to celebrate successes and people that have been involved with this enterprise for many, many years, in many cases unrecognized. Our small part in this imperative journey today is to truth tell, to listen to the narratives and discussion from a group of accomplished and intrepid pioneers who have all made substantial contributions to either the academy, the health enterprise, and community well-being, often with great sacrifice in spite of barriers, but with passion, compassion, and dedication across various stages of their lives and careers. We are honored to have them here, and I'm sure we will benefit from listening to and learning from their stories. In addition to these distinguished guests, I would also like to take a moment to recognize and honor our colleagues here at the Fralin Biomedical Research Institute who did yeoman's work in organizing and coordinating this program. And this is the organizing committee. I won't have time to go through and name everybody. You'll see those people around, but I truly want to express my deep gratitude and thanks for all the work these folks have done for the last several months on pulling this together. It just wouldn't happen without them, obviously. So let's get started. It is my distinct honor and great pleasure, but before I do that, I have to remember to put this slide up, sorry. I also want to acknowledge the incredible staff that we have, that in addition to the organizing committee, have been working very hard on this program. And you can see those folks here again. I don't have time to name them all, but it's an enormous effort to pull something like this together. So let's get started. It is my distinct honor and great pleasure to introduce the leadoff speaker at our Juneteenth program, Dr. Sylvester Johnson, who is the Assistant Vice Provost for the Humanities at Virginia Tech, where he also serves as the Director of the Center for Humanities and is the Executive Director of the Tech for Humanity Initiative. Dr. Johnson completed his undergraduate degree at Florida A&M in chemistry and education, minoring in religion, and subsequently completed advanced degrees, including his PhD in contemporary religious thought at Union Theological Seminary in New York. He held faculty positions at Florida A&M, at Indiana University, Northwestern University, before joining the Virginia Tech faculty in 2017. He's a giant in scholarship on the history of religion in the US, on African-American religions, and on race, religion, and colonialism. His award-winning scholarship is advancing new approaches to understanding the human condition and social institutions of power in an age of intelligent machines and other forms of technology innovation. And he's led a team of humanists and technologists to develop a successful proof of concept for machine learning systems that could assist in scholarly research. Whenever I hear Dr. Johnson speak, I learn something important. My eyes are open to see through a lens that has otherwise not been obvious to me, and I am moved to think more deeply about the topic of his presentation. What better person could there be to start off our program? Please join me in welcoming Dr. Sylvester Johnson.
Thanks. Thank you very much, Dr. Friedlander. It was a very kind and generous introduction. Thanks to all of you. It's great to see you again. And it's really exciting to be part of marking the occasion of observing Juneteenth. This gives us the opportunity to reflect upon, in recognizing the history of the celebration, what is a pivotal moment in the history of our nation and our world that could be described as part of the birth pangs of the multiracial democracy. Juneteenth, of course, is the holiday that is devoted to celebrating and observing the freedom of peoples who had been enslaved, approximately 4 million of them, and who transitioned from being dehumanized and treated as chattel to being recognized formally as members of the political community of this nation state. And as we think about the significance of this event, it's an opportunity for us to consider the relationship between multiracial democracy and the present and future of technology, in thinking about the prospects of scientific knowledge and research and innovation leveraged in service to shared prosperity. And as we think about this history of people moving from being chattel, from being in a democracy that had been marked by racial exclusion in a very foundational way, to transforming that into what some have called an experiment, others an ongoing struggle for an inclusive multiracial democracy, there are important ways that we can discern more fully what should be the relationships between what this occasion is marking and its social and political architectures, and what we're also celebrating and recognizing here today as the aim of achieving greater inclusion and participation in engineering and science. As we think about this history, we have to consider as well, in a strategic way, our future. And so I'm going to offer just 3 quick points that we might bear in mind as we are engaging with the other panelists and speakers and having our conversation today. And the first is that the future of humanity is human engineering. And I don't mean that in a reductive way. I don't mean it in a simplistic fashion, that there will be nothing else. And I certainly don't mean it in a very utopian, uncritical way. But if you think about some of the recent developments that we have seen in our capacity to alter our humanity, just January of this year, a biotech company actually based in Blacksburg, Virginia, led the effort to transplant a heart that had been grown in a pig that had been humanized into the first human subject to receive this cross-species organ transplant. And that patient, who was a terminally ill cardiac patient, was able to live a couple of months with that transplant. Our ability to actually change the nature of this pig to something a little bit more like our human genetic makeup is just the beginning. It's the portal of entry of what will be an explosion of capacities to be able to sustain human life in ways that are very creative, that will not come without controversy, but that are certainly witnessing our ability to alter ourselves and other organisms. Even thinking about way back in what I call BC times, so before coronavirus. It seems like it was so long ago, but pre-COVID times, the world witnessed both intrigue and outrage over the work of the Chinese scientist He Jiankui, who actually genetically modified embryos with the intent of making them inherently genetically immune to being susceptible to immunodeficiency virus, or HIV. He violated multiple standards of ethical conduct that existed in China and globally, particularly because he was altering heritable characteristics. This was a stem line genetic engineering, which is not supposed to be done. However, above all of the outrage and intrigue and condemnation, he lost his job, he was imprisoned. He actually just got out of prison in April of this year. I think it is also important for us to consider that there are billions of dollars and our best intellectual human capital being leveraged in order to make it possible for us to modify our DNA, not just to cure people with somatic cell lines that cannot be inherited, but also the same technology that can be applied in order to do something that perhaps one day, for instance, might be necessary to preserve our species from extinction by who knows what kind of pathogens, since we are currently experiencing a global pandemic. In other words, I don't think we should let the outrage and the abysmal failures ethically and morally of that incident distract us from the fact that the massive amount of capital that is going into make it possible for us to engineer and to re-engineer at the level of our cells and our genetic constitution, what it means to be human. We should not allow those things to distract us from the fact that we need to get ready for the day when our ability to do that will become so good, it will become so reliable that we will be able to undertake stem line genetic changes that can be transformative as we attempt to try to preserve our humanity and our lives. All of that is an example of some of the changes that are going on. What is called often the fourth industrial revolution is seeing the growth of the ability to combine human systems and what we often think of and call machine systems for brain-computer interface technologies, or BCI architectures, that have tremendous applications, including restoring capacity to people who experience traumatic brain injury and also other things. But the point is that the future of humanity is going to be very challenging. And rather than seeing these things as blips on the screen that are anomalous, I think what we have to do is understand that we have to prepare ourselves socially and politically for the day to be able to guide the access to and the application of that capacity and that power in ways that are going to get us shared prosperity. The second point I want to make is that public interest technology is one of the most important emerging and growing fields that our nation and our world is seeing. In Europe, this has been referred to more as responsible research and innovation, or RRI. There's lots of terminology globally that has been used for this, but the point is that in order for us to advance and to be able to achieve the kinds of small d democratic outcomes that we need in our world, we have to actually invest in new approaches to understanding what it is that technology is demanding from us and who should be participating. And I use that first example of human engineering as just one provocation to think about the second point for public interest technology. So what is public interest technology? This is an emerging field that essentially is devoted to making public interest and social good the most important factor in how we try to understand what technology is and how we identify who the participants should be, of what it means to be a technologist. If we pay attention to the fact that the greatest debates that we are having about our science and scientific and technological innovations and research and production of new knowledge, the greatest debates, what is most difficult for us to resolve, are not what might be called the technical dimensions of it. And those things are very difficult, and they are very challenging, and they are very important. But we're actually much better at solving technical challenges. We are. We can put people on the moon. We can actually manipulate a rover autonomously, or rather remotely and autonomously on Mars. But we can't figure out what to do with privacy. We can't get the design of our algorithms to function in a way that does not target some above others. In other words, the humanistic ethical dimensions of these things, the ones that are concerned with what it will mean for our society, have to and are becoming part of how a new generation of technologists are shaping and being prepared to create the future talent for our society and our world. This is happening nationally and globally, and within the US, what's called PIT, or public interest technology, has been emerging in a very vociferous way to get us ready for this challenge. The last point I want to make is that just as the history of Juneteenth has been concerned with this democratic, some call it experiment or struggle or commitment to make it possible and to operationalize broad participation in the governance of human beings who need to have the kinds of access to rights and civil liberties and human rights that are essential for human flourishing, so also are we going to have to reckon with the need to bring democratic governance to technology. And that governance of technology has become a point of concern across our world. We're seeing it play out in the kinds of efforts that private entities and private corporations, for example, have begun to devote to the creation of ethical boards. We've never invested in the talent that we need to bring democratic governance to technology in the past, because we defined that in a way that excluded the multiple range of actors who needed to be there across disciplines. And so what we have to do now is understand that just as our democratic struggle and experiment has required us to move from a model of racial exclusion in order to bring people into the political community of those who are members of the state, who are able to participate fully in society. Now we're going to have to do the same thing with our scientific knowledge production. So this means the integration around projects and problem solving, and the expansion of who and what a technologist is, in order for us to be able to bring governance, democratic governance, to technology. When I say governance, I don't just mean government, I mean just managing it. Who's going to create it? Who's going to participate in employing it? And how are we going to move in a way that gets not only our local societies and our nations to deal with the problem, the looming problem of inequality and unequal access and unequal participation, but actually our world. The kinds of divides that we'll hear about today historically and that are current are not just regional and nationalistic divides, they're global divides. And we have this global community. And so I leave those 3 points with you because I think what they offer for us is not only an opportunity to reflect upon, more fully discern the relationship between the history and ongoing struggles for multiracial democracy on the one hand, and our scientific knowledge production and our technology on the other, but I think they provide a possible roadmap for us to be much more strategic, much more visionary, and much more deliberate in structuring the kinds of architectures for disciplinary participation, for connecting the different sectors of our society, whether it's private entities, whether it is public institutions, whether they're civic organizations or governmental organizations, in order to move us in a way that we prepare for our human future, that we're advancing public interest in our scientific and technological discovery and innovation, and that we are creating the right kinds of preparation of future talent and relationships across sectors of our nation and our world to bring us to the point of democratic governance of our science and our technology. I want to thank you again for the opportunity to speak with you. I'm really looking forward to hearing the different panelists and speakers we're going to have with us today. I want to thank all of you who are here at Fralin Biomedical Research Institute for taking the wonderful step of making this reflection upon our nation's history and our world's history integral to how we are thinking about the mission of our institution today and how we're advancing technology and science. So thank you again for the kind invitation.
Hello all. My name is Monet Roberts, and I am a postdoctoral research associate in Dr. Jennifer Munson's lab here at FBRI. I want to take a note to let everyone know that we are going to try to be mindful of time to give all of our distinguished guests time to speak. And so we're going to strive to stay on time as much as we can. But without further ado, it is my distinct honor and pleasure to introduce Dr. Manu Platt, who is a professor at Georgia Institute of Technology in the Wallace H. Coulter Department of Biomedical Engineering and principal investigator of the Platt Lab for Tissue Remodeling, Repair, and Regeneration. Dr. Platt attended Morehouse College, where he majored in biology during his undergraduate career. And he was also mentored by the late and great Dr. Bob Neuron. He then went on to pursue a PhD in Dr. Hanju Zhou's lab on endothelial cell biology. And then he did his postdoctoral work at MIT before joining the faculty at Georgia Tech/Emory Joint Biomedical Engineering Program, where he investigates the most potent mammalian collagenases and elastases known as cathepsins in various pathologies, such as HIV-mediated cardiovascular disease and stroke and sickle cell disease and cancer. His work reached far beyond the lab, where the translational aspects of the lab was explored in patients in Ethiopia and South Africa, as well as within the US in investigating policy and the governmental response to HIV/AIDS and impact on affected communities. He continues to do not only great scientific research, but touching the lives of so many through being the founding director of Engaging New Generations at Georgia Tech through Engineering and Science, or Project Engages, which is a biotechnology and engineering research program for African-American/Black high school students. He is a champion for diversity, equity, and inclusion, especially in the academy through many lectures that he has given across the country. He has received numerous awards for his efforts in DEI mentorship, specifically the AAAS Mentor Award in 2021, which I proudly nominated him for. One of the Route 100, and among other many accolades. Please join me in welcoming Dr. Manu Platt.
Now, see, they want to keep us on time, but they got to limit these introductions. No, thank you very much for that wonderful introduction, Monet, and thank you for so many things as well. Thank you all for having me. This is exciting to be here. Happy Juneteenth. And this is actually a big deal. This is my first Juneteenth talk, right? So many firsts that are happening. Let me go ahead and get this set up here. And I do want to thank the organizers for having me and for dealing with me in my email response times as well. But yeah, very excited to be here to have this discussion with you today. And I will be speaking again on this Echoes of Juneteenth, which we know is this holiday where there was freedom delayed, but then how that maybe is still pervasive today and healthcare innovation denied. But really hopefully one of the messages that I hope to give, particularly to the young people in the audience, is about how you can take a career even in biomedical science and still broaden it out to impact so many different areas instead of just thinking of, I must just do this one thing that I was told to do. All right. So again, we all here know why we're here about Juneteenth, and that when the Emancipation Proclamation was signed, the news did not get into Galveston, Texas until 2.5 years later. And some of these early slides were prepared by my wonderful sister-in-law, Aziza Platt, who you will hear from later. But a Juneteenth miracle has already happened, if you had not heard. Beyonce is releasing another album July 29th, and it was just announced. Juneteenth being a federal holiday has already been a gift to us all. It will be called Renaissance. And I love this tweet that tells some of the other Renaissance painters, watch out, because Beyonce now owns the Renaissance. So this is just enough of a reason to celebrate Juneteenth. But the rest of Juneteenth is beyond just this message that got to the enslaved peoples in Texas. There was these very times during which people found out about emancipation. And this one in the middle actually really surprised me when Aziza shared this fact with me, because I went to high school in Dover, Delaware. And to find out, you think Delaware is one of these northern states, but that they only ended slavery after the ratification of the 13th Amendment, and that there were still battles in Texas with Supreme Court decisions through 1868 and 1874 even, before full emancipation was reached. And a little known history fact that there were the Muskogos, descendants of Black Seminoles, who escaped from US slavery in 1852 to Mexico, and they also celebrate Juneteenth. So there's way more parts of this story that I think will be unraveled as we now as a nation begin to celebrate this holiday. And so again, why celebrate? It's to honor those people, our enslaved of African-American heritage, but also to acknowledge that because of these continued systemic racism and inequities, police brutality, mass incarceration, here I'll be talking about health disparities, redlining, et cetera, slavery mutated into different ways that still Black people are still fighting for full liberation. Again, shout out to Aziza. And so that's why we want to actually be talking about this. And I love that you brought Juneteenth into a scientific format. Yeah, coronavirus hit. And America had to wake up. Others of us were already awake, but realized some of the massive health disparities that exist in this country. And we know when the coronavirus epidemic or pandemic first began in the 20s, it was it was impacting Black and brown people, and that's who was dying at first. Then once that became a realization, they said, hey, let's open up. I'm not dying, it's those folks, right? And so then once they then said, oh, well, let's open schools again, because we need to get this economy going. Kids don't get it, no problem. But then the children that did die, though they weren't in the same numbers as adults, were what? They were Hispanic, Black, and Native American kids dying of coronavirus. And so I went to the AIDS 2020 virtual conference. It was supposed to be in Mexico City. They made it virtual, obviously, because of coronavirus. And Dr. Celeste Watkins-Hayes, who's also a Spelman alum, love that. She gave this talk and she said, people say we're all in the same boat. It's a disease, it gets to everybody. But we know people were dealing with coronavirus in different spaces. Some people were trying to survive in a little dinghy boat, and others were living in these massive, excessive yachts. It wasn't the same for everyone. And even to the point that as we've done more work and more of the analysis, I love this article. I don't love the article, but it's a well-written piece about how COVID-19 hollowed out a generation of young Black men. Black men or Black people, the age 35 to 44, my age group, were dying at 9 times the rate as white people of the same age. And it was greatest in Black men. This John Henry syndrome of I got to go out and make the money for my family. I've got to put myself at risk to feed people. But we know the chronic health issues and other things that we in marginalized communities experience made us especially vulnerable to death due to COVID-19. So whenever I think about Black men, yes, I'm a Black man, but I'm also have 5 Black men that are my brothers. I'm one of 6, I'm the second oldest. And I didn't know about Juneteenth until summer of 2000, when I was interning in California. And there was this Juneteenth celebration in Oakland that we all went to. Because I'm originally from New Jersey, born in New Jersey. My dad was in the Air Force. We went to Arkansas for a little bit, then Oklahoma. And as I previously mentioned, I did middle school and high school in Dover, Delaware. And then I came down to Morehouse College in Atlanta, Georgia to get my undergraduate degree, a bachelor's of science in biology. I knew I wanted to do biomedical engineering, and they had a dual degree program that I was really excited about. And the reason that I chose Morehouse over the other institutions that I was interested in is I visited the campus and there were Black men everywhere, going to class, being polite, saying hello. It just fractured everything that I had been growing up in the 80s and 90s hearing about Black men. I was just shocked on that campus, and that was it for me. And I said, I'm going, okay? And I really learned a lot about myself then, because it also shows you Black men are all different types of people, right? And so when you go to a place where it's just Black men there, you don't have to worry that people are gonna put Black men stuff on you, and all the other parts of you can begin to come out and shine, right? And I met some of the greatest friends of my life there that are still my friends. I also met, again, my sister-in-law, who's my best friend first, Aziza, who's a Spelman alumni there as well. And so in that framing and beginning to really think about how Black people do matter, Black people can do science, Black people can do anything, I, out of my friends, continued on this academic route, got a PhD in biomedical engineering and opened my own lab in 2009 at Georgia Tech, as Monet mentioned. And the things that I begin to study and care about are things that are racial health disparities. So I'll talk about sickle cell disease. I'll also talk about our work with HIV and cardiovascular disease. And then I do a few other cool things in breast cancer and other things as well. But I think it's important for me to be in this space and to then study diseases that impact people that look like me and that are usually understudied from those lenses. So I say I run this lab on, we've studied tissue remodeling, right? And so I like to make sure the audience is aware. So tissue remodeling is how do you go from this healthy tissue, this is a human coronary artery, supplies oxygenated blood to the heart muscle, to this atherosclerotic plaque that, as you can see here, the lumen is this open circle here, so blood is passing through there, but you can see that's fully, not fully, but almost completely closed off here, which means this person was great risk for a heart attack. And we actually got this from a person who had undergone a heart transplant, so this was the bad heart. But for this tissue to get remodeled, there's some initial degradation of the structural proteins in the healthy artery that then causes the cells to receive different cues, migrate into the space, lay down new proteins, and you start to get this process of remodeling that tissue. Now, where biomedical engineering comes into play is it's not just the cellular or the biochemical properties that are changed, it's also the mechanical properties of that tissue, and the mechanics feed back in with the biology in many systems. And so these are things that we want to understand so that we can stop these processes. Most people know about osteoporosis. You get this dense collagenous bone matrix, and as it is degraded down, you lose the mineral component and the collagen component, and you now get brittle bone. So again, not only changing the biochemistry, you're affecting the biomechanics. And this little guy walking is this cool biobot, part of this NSF center called EBIX that I was a part of, taking skeletal muscle, seeding it into a matrix gel, fibrin gel, allowing it to mature with these PDMS feet, that if you applied electric voltage to the media, the muscle would contract and the bot would walk across the bottom of the plate. This was designed by my colleague at Rashid Bashir, University of Illinois, but our labs group came along because these bots would just break in the middle as they were walking. So we want to understand what are the enzymes that are being expressed that cause it to break so you can design or engineer a functional time of this bot. But at heart, my lab studies things falling apart, okay? Morehouse College is a great liberal arts education. We read this my freshman year in World Lit, and just an important story. So you'll hear me apply pop culture and literature and other things. But who are these buggers? So the agents that I really focused on are these cysteine cathepsins, as Monet said in the introduction. They are forever engraved in her brain as well. And they're so important because they are the most powerful collagenases and elastases. That means they can degrade this major structural protein collagen in our bodies better than any other enzyme that we have. And elastin, best named protein, it provides elastic recoil to tissues, right? They can also degrade that better than any other enzyme, but they're generally understudied because they're kind of difficult to work with. They prefer an acidic environment where we think in biology that once you get outside of the cells, most things are neutral. People used to think that when I started grad school. Now we know in disease states, environments get locally acidified by a number of other mechanisms, making it perfect for these enzymes to degrade that tissue and remodel it. And also big pharma has been trying to find inhibitors for these cathepsins because there are several tissue destructive diseases in which they have been highly implicated. But out of 16 cathepsin inhibitors that have made it into human clinical trials, 16 have been denied. So think that is billions of dollars. That stresses me out, but the last big one was Merck, and they ended up laying off that whole division of people once it didn't make it through. So we need to find ways to get one or more through. And so when I first started my lab, we had these two main hypotheses. First, because they're so hard to detect, we thought maybe improper quantification was causing people to dose inappropriately with these inhibitors, causing these problems. And then later on, we began to realize there's some other underlying functions of the biochemistry and physiology that we still don't understand. So we wanted to take a dual approach. And so again, I won't belabor you this, but we love this thing that we developed, this technique called Cathepsin Zymography, to detect the mature active form, because that's what you want to target with a small molecule inhibitor. Based on the principles of SDS-Page, where then the enzymes actually are refolded inside of the gel after being separated, then you activate them and they cleave the substrate in situ, such that you can stain the entire gel with Kumasi blue and get a cleared white band where there's not the substrate protein, showing where the enzyme was active. Why this is so great? There's no antibodies. Many people here do Western blots, right? And these other things. There's no antibodies, which saves so much money. We take antibodies and pour them down the drain. And the low cost nature of this assay is what allowed me to do work in developing countries, as Monet mentioned. And so when we started developing the zymography, we also found out it was more sensitive than Western blot because there's a bit of an amplification of the signal. And then we could multiplex it for these four main cathepsins that we study that are really involved in the tissue destructive diseases that I care about. They're denoted by letters. So cathepsin K, L, S, or V are different gene products. So they were identified in cancer cells. And so that was our first application where we got our hands on patient-matched breast cancer samples. So normal and tumor from the same woman. And we loaded the standard curve of cathepsin K, recombinant enzyme. And I love this. So I teach engineers, right? And engineers want things to be binary. As we know, biology is rarely binary. So that's like this break for them of like, it can be in the grayscale. But then we did this and it was binary, right? Shut me up. So this is from the same woman, normal tumor, normal tumor, normal tumor, normal tumor, normal tumor, off in the normal mammary tissue, on in the tumor tissue, such that we could do absolute quantification and could almost draw a line to differentiate normal versus tumor tissue. And we got 100% sensitivity specificity. One of the earliest papers out of my group, we then profiled lung cancer, cervical cancer as well at different stages. Bin Chen was my undergrad actually who was doing this work. And we thought, oh, great for breast cancer detection, but that wasn't the biggest problem, right? And so we then began to use it in other ways of using it for personalized medicine, because how can you then understand why if two women have the same type of breast cancer, one will have a more aggressive form? So you can kind of help a woman make a decision with her and her doctor about, do you go for the prophylactic double mastectomy, or maybe you could benefit from a more conservative treatment that you are very watchful of. So those are things that we've been continuing to develop. But these cathepsins are in so many other tissue destructive diseases. So we look at them, I'll talk about sickle cell, our HIV work, tendinopathy damage, and also even endometriosis, which is a woman gynecologic disease that is heavily understudied as well. But then my mind kind of, again, the HIV work, my mind blew up when I was starting my lab and there was a call from NIH and the International AIDS Society to bring new investigators to HIV work, because the virologists had done a great job with the antiretrovirals, but they found out people were dying of other comorbidities. Cardiovascular disease was one of them. I study cardiovascular disease, and it would come with a free trip to Cape Town, South Africa. Okay. Yes, I had never been to the African continent before, and I applied and proteases were involved, and I was selected to go, and it really just, again, changed so many things. So they took in 22 investigators from around the globe. Only 6 of us were from the United States. So you can see my big head there. And Denise Evans would end up becoming a long-term collaborator with me. She's at University of Witwatersrand in Johannesburg, South Africa. So they were giving us HIV from rooter to the tutor. That was the first time I met Dr. Fauci, and Dr. Francois Barré-Sinoussi led off the meeting. She had just won the Nobel Prize for identifying HIV as the causative agent of AIDS months before this meeting. So we're in the room with this Nobel laureate, right? It's amazing. And one thing that she said is when they were first, this is before HIV turned 30, right? We just passed its 40th anniversary last year. And they could not grow the virus in the lab. And she was working at the Louis Pasteur Institute in Paris. And she said what they had to then do is throw everything they knew about human lentiviruses out. And she said to those young investigators, don't start with dogma. You'll never hear that in a lab, right? Forget about the literature. But we had to almost do some of that with COVID-19. And there's so many interesting parallels. So that don't start with dogma meant we are free to break barriers and break into new spaces that others may not be thinking about. She had a Nobel Prize, right? By doing that. And so that became a mantra I would do in my own science. But what also changed my viewpoint about being a scientist is there were always activists and protests at the World AIDS meetings. Organized protests. So when they would have the plenary would be about trans healthcare access, the trans groups, activist groups would take over the stage and have a protest. And then we'd all learn about it from the activism side as well as then the science side and the policy side. And it made so much sense when people would tell us because those early HIV medic or doctors, they had to fight for their patients. There was stigma, people didn't even want them in hospitals. And so the people who were doing the care and the research had to step out of their lab boxes and actually become activists. And so here you see the mantra for that year is right here, right now. This year, the year I also went in Vienna, they had a march through the streets of Vienna, Austria, ended at a concert in the courtyard of a castle. My sign here says criminalize hate, not HIV. And they had the AIDS ribbon on the Austrian parliament. This is before the United States ever did that. So walking through the streets, I was blown by what they were doing. And I was actually marching by the people with the red umbrellas. And these, I found out later, were the sex workers. And the sex workers, as we were marching, their chant was sex work is work. Sex work is work. And I think now we hear that more spaces, but it becomes important of solving an epidemic, right? You can't exclude people and say, oh, they're sex workers or they're prostitutes or they're this, we won't give them medical treatment. The disease is still going to spread because they have customers. So that's where you have to, this HIV AIDS community has to break out of stigma and all of these other things you think about to actually solve the problem. And so again, I really began to take on this idea of being a scientist activist. So when I used to come home and watch my after school special programs, they would say things like, think globally, act locally, right? That would be the message. But then the world shrunk with international travel. So then I said, act globally, right? And I was this HIV AIDS community, was this global community. But another reason that became important, I was at these AIDS meetings and they were talking about all these other countries and these high rates, but the rates they were, and I was like, what about the US? And they're like, the US, yeah, you have low rates. But the rates they were talking about in these other countries were the rates of Black and Latino populations in the United States. Why are we not talking about these? These are by communities that we should be concerned about. So it's that global health concerns are health disparities for underrepresented minorities in the United States. And we should think about that and then understand the importance of that, becoming a scientist activist and studying health disparities. I might be able to go to a Black community and Black high school students and talk about HIV AIDS. There's other people may not. And I can talk about it from a science perspective to debunk myths, but also then connect on other perspectives that maybe they would hear that message. And I also say this, especially for Black and Brown communities, it's almost funny like we have to out ourselves as scientists. Because usually we're the only scientists in our group of friends, unless you're with your science group. And then you're like, oh, we don't have to talk about what I do, I know. Out yourself because they might have questions for you and might want to know that. And I have to do that on planes a lot when I used to talk to people on planes. When they were like, what are you, a professional athlete? It's so funny always. No, but I'm a scientist. And again, things that affect our communities. Dangerous disproportion. 2011, white men who have sex with men had the highest rate of new infections. Right behind them were Black men who have sex with men. Remember, Black people are 13% of the US population, Black men 6.5%, but look at how close these numbers are by absolute numbers compared to white men. But then here's the other terrible statistic. Black heterosexual women were right behind that. And again, that's why stigma and stigmatizing men who have sex with men or DL or whatever doesn't get people the help and stop the pandemic or the epidemic of HIV AIDS. And you can see Hispanic and the rest. But by 2016, look at the flip. Black men have sex with men, the numbers didn't change so much, but the number of white men who had sex with men that were contracting new HIV infections dropped significantly. And so the annual number of HIV diagnoses actually declined by 5% between those years, but you could see where the decline happened in the white population. And what happened? Access to drugs, access to pre-exposure prophylaxis, reduction of stigma around PrEP versus in Black and Hispanic communities where there was a lot of stigma. Now I live in Georgia, which is the darkest state on this map, also home of the CDC. And so these are things that affect my community. And so what I learned at that first meeting is that people with HIV were at increased risk of heart attacks or strokes. The questions were, was it inflammation due to the virus itself? Of course, or side effects of the highly active antiretroviral therapy. And so we joined the fight, and we came up with a number of wonderful mechanisms. And there may be a familiar name on here. Fralin's own Monet Roberts, as an undergraduate in my group, was on 2 of our HIV/AIDS papers. She's been a star from the beginning, so you're lucky to have her. But this is what took us to South Africa. And it's funny when people say, oh, you've been to South Africa, that's so cool. Well, I couldn't get samples from Emory in the US because they thought what I was trying to do wasn't worth them giving me samples for. That's fun. And so Denise Evans, who I highlighted in the other picture, she was like, if you come to Johannesburg, we have 400 patients a day. You're welcome to have the samples. Boom. So we began to fly to South Africa, taking our cheap method and technology with us that we could set up in the labs there. There's my colleague, Rudy Gleason, who's a Georgia Tech professor, who I do global health with also. I am nervous. This blood has HIV. When you all do your bloodborne pathogens training, you're supposed to treat it all as though it may have a disease. Yeah, this one does, okay? I look back, I should have had on goggles. I'm very clear now about my PPE was not fully correct. But also while I was there, you get to experience the culture and see the things that are offered there. And what we began to find is that our cardiovascular hypothesis, we thought there would be increased protease activity in people on these antiretrovirals. And in the first Zymo we were doing, MOP is me. So we thought that the higher the signal, the more dangerous it was. I had the highest signal on here. That was scary. But it's also important to remember your hypotheses can be wrong. We test hypotheses, we do not prove them. And it turned out that the patients in red were all on antiretrovirals. So they did not have the increased activity. But what surprised us is that it wiped out this protease activity in their white blood cells. We weren't expecting that. And this is the importance of my international collaboration with Denise, and that there was this one patient who was prescribed antiretrovirals, but still had a signal. And I was like, oh man, our cardiovascular hypothesis, forget it. She's like, you know, May, this is quite interesting because we're trying to find a way to monitor if patients are adhering to their antiretroviral therapy. Dumb me, I was like, why? If they know it's gonna save their life, they've got to take it. Why do you need to monitor it? Because people don't take their pills when they're sick, but also the risk of generating drug-resistant viruses that then there's no regimens that they can then manage down. And if you just counsel them and explain why they need to stick with it, then they will start taking it. And I said, but how do you detect it now? They use mass spectrometry, which is expensive, right? So of course they don't even use that in the United States. And they do also surveys and questionnaires. So she then had this idea, we can use this assay as a way of monitoring adherence to drug, almost like a biological lie detector test. Oh, great. And again, thinking about ways in low resource and low cost settings because of working in that environment. So this is the first prospective clinical study that I've ever done in my career that led to multiple trips back and forth from Atlanta to Johannesburg, where we were collecting samples of patients when they were first diagnosed, and then after 6 months and 12 months of being on antiretroviral therapy. And what we found is that, yeah, at a person who's antiretroviral naive, you could see some didn't have any signal. There's a wild fluxes in their immune systems. But after 6 months, you could see a significant reduction in the signal there. And we were correlating this with their viral load. So if there was an undetectable viral load as the gold standard of they are adhering to their medicine. And so we got great specificity. Denise is an epidemiologist. We were looking for the negative signal, so I want to call it sensitivity, but she says because it's the negative of the negative, it's the specificity, so I'll go with that. But still, this is quite exciting, and we're trying to find ways to put this on a chip and make it more accessible, because if you want it used in the rural clinics, then you need to reduce some of the technological requirements and the protocol. But with Rudy Gleason, this also led us to work in Addis Ababa, Ethiopia. He had relationships there. So he was like, hey man, we can do the testing here if you can send a student. Rudy's from Texas, that's why I talk like that. And that's actually the emergency room at Blackline Hospital. And so I sent my student, Ivana Parker, who was Monet's mentor, and we supplied the reagents for this lab and took our cheap assay there. And Ivana was really the one that got it working there. And we began to optimize it because it's really comfortable in the US with all of our stuff. But each lab we went to, we had to optimize it for different things. For example, in Ethiopia, they shut the power off in the building every night to save money. So Ivana would go and get her samples and the cells and the boxes would be soggy and like semi-frozen. Our enzymes lose activity every time they thaw. So therefore that became an issue. Plus we can't keep a water bath on all night if they shut the power down and if we were incubating at 37 degrees Celsius, yes. So what we did is we optimized this assay. She had to do it with fresh cells the day we drew the blood, and then you could incubate it at room temperature overnight and get a detectable signal. And that's what we had to begin to do. But of course, I had to go to sample the local nightlife, and Ethiopian dancing is amazing if you've not done it before. And yes, they put me on stage because I am a star. You're welcome. But I'm really proud of the paper we then published with my South African collaborators, my Ethiopian collaborators, to really start to talk about how we could monitor this with these techniques. And again, with the HIV/AIDS community, not only was there activists, but also policy and interfaces with government. So this is when it was finally in DC in 2012. This is a big deal because the US had terrible visa laws. If you were HIV positive, you could not get a visa to come into the United States until 2010. Barack Obama removed that law. So the AIDS Society said, we're not having a meeting in your country until you drop that rule. And at the time, Washington, DC was the highest HIV infection rates in the country. The capital was DC proper, the actual city. So Hillary Clinton spoke at that meeting. And so there was all of these policy issues that I began to hear. And one of my younger brothers is a political science professor. And I used to say, Matthew, we should really do something about the science with HIV, with the policy and the government's laws being passed. For those of you that have younger brothers or sisters, you know, they're ornery and resistant to everything. You're like, no, no, no. And my parents would be like, Manu, leave Matthew alone. What? What? Anyway. But I want to shout out Ivana, excuse me, who's now an assistant professor at University of Florida. But so finally, after year 3, I convinced my brother to work with me on this paper. And so we were able to actually look at how the relationship of the scientific publications around HIV/AIDS may have any correlation with laws that were brought up in Congress and or passed. I had the hypothesis that our Congress people listen to the science since we're federally funded by the government. My younger brother's like, why would they listen to scientists? Of course they don't. Anyway, he was kind of right. Except there were some issues that did directly correlate with what we found out about the science. So it was great to publish this with my younger brother, who's now a professor at Morehouse College, our alma mater. So again, I told you about that think globally, act locally, and now I want to say act locally. Oh, okay, really? Okay, okay. So just quickly, I'm going to blitzkrieg through my sickle cell work. Sickle cell still is a problem. There's genome editing solutions that people are working on, but it still is killing predominantly African Americans in the United States. You can see the life expectancy there. But again, it is a global health problem. That is a health disparity where up to 90% of children in the global concern will die in their first 5 years of life. And this is pretty terrible. So I'm going to skip through the molecular origins because Monet is itching in her chair. But there's this pain crisis that happens. These red cells that sickle, they block the blood vessels. And the issue with us as scientists and medical people is we still can't quantify pain, right? So they live their lives in pain. And then when they go to the hospital, it is extreme pain that we probably cannot imagine. And so again, this is where medical racism comes into play. They're treated as though they're drug seekers and you just want painkillers, but they actually have this chronic disease that causes pain. And even such that nurses thought that African Americans with sickle cell were coming in to seek drug addiction because they were addicted to opioids, but it actually is not true. The number's even lower because this is one of those medical racism things that help Black people because they think our thick skin means we can handle more pain. They would not prescribe Black people opioids as they would other populations. So the opioid crisis is reduced in our communities. But this is a picture of an artist with sickle cell about what a 10 feels like. They say on a scale of 1 to 10. And this is what a 10 feels like for him with sickle cell. So I care about strokes in children. Again, I'm gonna zoom through this because I want Monet to have a fit. But not only does it cause paralysis, but there's also these cognitive strokes in thinking parts of the brain that aren't visible. So then you've got this Black child sitting in class and they're getting bad grades and like this little lazy child, ooh, they're having strokes. And so this is why we need to be monitoring this and watching it. So we're looking at some new mechanisms. Again, I'm gonna skip through this because today's itchy, sorry, thought I had more time, okay. But we actually have a mouse model of sickle cell disease that we've been looking at new drug targets. We've been studying this clinically since 1910, okay? One good drug on the market. There's another 2 that are coming out now. We know the genetic mutation, the protein, molecular nature, one drug. I'll let the audience think about why that's a real thing, okay? So we've been looking at new targets. Our cysteine cathepsins are involved. We found a drug molecule that inhibits a signaling pathway that would turn it up such that we could block it. And in doing that, we also can protect the structure of these arteries and the mechanics of the arteries to prevent those complications, all right? And I also work with the Sickle Cell Foundation of Georgia because again, by talking to the community, you can learn more about how this disease works, how it impacts people, and they run their own tests on themselves all the time with diet, exercise, water intake, and they can tell you a lot about what we can then go test the mechanisms in the lab. And also we are activists with them. We petition the state government when needed. I'm on their board of directors now, but I also use it to teach in my classroom because if I'm training the next generation of engineers, let's get some really good ideas about sickle cell from you all, and you all can begin to think about it instead of waiting till you become a professor. So just with all of that, yes. As a professor at an R1 institution, what? Yeah, it's a lot. And then especially when I started my job, they sent this out, the NIH professors who are Black are 50% as likely to be funded. That's awesome. That doesn't scare you. And that also certain topics are what drive these funding decisions, and things that are racial health disparities are some of the topics that get scored poorly. And I'm out here studying sickle cell disease. What? And peer review. Every my colleagues say, it's not me who's scoring the Black people down. It's not me who's doing it. Well, so I wrote this article last year, we exist, we are your peers, and we also see you on study panels. But still, to push forward, it takes courage. And I love this Maya Angelou quote, and she says, courage. The most important of all the virtues, because without courage, you can't practice any other virtue consistently. So to the young people that I was talking to last night and those in the audience, if you're scared of this career or all these other things, courage, we're out here, right? And we can help support each other. And that last bit, even pre-tenure, started this high school research program with Bob Neer, one of my mentors, and Mr. Vance to bring Black students from Atlanta and to work in Georgia Tech labs. And we pay them, we were paying them more than minimum wage. Minimum wage in Georgia is $7.25 an hour still. We used to pay them $10 an hour. Last year, we went up to $15 an hour. So we got the best job in the city, right? If you don't have a high school degree. Project Engages, we are now going into our 10th year. And actually the reason I need to be back in Atlanta tonight is we're actually having a 10-year reunion. We have touched over 140 students. Many of them are first-generation college students. They've now gone on to college. Now we have plenty of graduates from college and a few that are actually in graduate programs now. So this is quite exciting, all of this while doing these other things. And I just want to say, as you move forward, reach back, pull someone else up, but celebrate them being themselves, right? Because what we thought we're told scientists look like means that's not for me. But I look like me and do science. I got dreadlocks, I got earrings, I love hip hop and R&B. But that also means you should bring your whole self as well. And I incorporate this with my lab. There's a young Monet teaching us how to do the early wobble, I believe. And so with that, thank you for listening. I look forward to conversations and the other talks from today. And this is actually from my 10-year lab reunion from a few years ago. And you can see that the impact you can have besides the biomedicine, but also on people that then go out and start their own groups and propagate those successes. Thank you all very much.
Okay, all, I think we have time for one question. We have mics right here on this row, so you can come up to the mic and ask your question. Okay.
Yes.
Can you hear?
I can
Okay.
hear you.
So you started by saying there was a huge disparity in African-American men in a relatively young range in terms of COVID deaths. And I was wondering, is that sort of biomedical, something to do with our different chemistry, or is that something to do with just treatment and access to
No, that's a great question. And so there's several, all of those things, right? But it was really that they are in those frontline jobs. They could not stay home and work from home. So they were then going out into the world, working these exposed jobs, especially in working plants and factories where there was no protection offered. And so they were this John Henry thing about, I've got to be the one to make the money, even though they also have chronic health disease, high obesity, diabetes, high blood pressure, but still going out to work. And we know the reason they would have the high blood pressure and the rest because of the socioeconomic environments and diets and the rest. So it was all of that together, and being out there with COVID was what was killing us. Yeah, thank you.
Awesome. Well, let's thank Dr. Platt once again.
Take this over. But hello, everyone. I'm Audra Barnes, a first-year PhD student here in Dr. John Chappell's lab, and I have the honor this morning of introducing Dr. Kayla Singleton. So Dr. Kayla Singleton is a postdoctoral fellow at Emory University in the Fondes lab. She is also a NINDS DSPAN scholar and a Burroughs Wellcome Fund postdoctoral enrichment fellow. Kayla received her bachelor's degree in neuroscience and classical history from Agnes Scott College and a PhD in neuroscience from the interdisciplinary program in neuroscience at Georgetown University. In the Fondes lab, Kayla investigates mitochondria integrity and localization in Menkes disease. Kayla is also the co-founder and president-elect of Black in Neuro. Kayla has participated in numerous speaking and panel engagements. She has numerous publications, and she has numerous presentations, including invited seminars, posters, and selected talks. Kayla has taught 9 courses, serves on 9 academic committees, and is a mentor to 20 mentees. Kayla has received numerous awards and honors, including Forbes 30 under 30 class of 2022, Cells Mentors 100 Inspired Black Scientists, and Go Magazine's 100 Women We Love. Kayla has also been featured in several media outlets, including National Geographic's Real Scientists, Nature Medicine, and NeuroCentral. Please join me in welcoming Dr. Kayla Singleton.
Hello, thank you all for being here, and thank you for that really great introduction. So today I'm going to talk to you about really how I approach science and the way that I feel like my experience has shaped what I do in research, and how I think both like my personal identity and my professional identity have really been enhanced by my community, but also by the research that I do. And so to start, as a developmental neuroscientist, I always like to start at the beginning. So these are my parents. This picture was taken, I think, in 1994, so I'm like 2 years old. And I grew up in Grayson, Georgia. My parents met in the Army, and due to some like frowned upon fraternization, they got together and then they had me and they left the Army. But I grew up in this affluent suburb, Grayson, Georgia, which like the picture of suburbia, just like shopping malls everywhere. And growing up, I only had a couple of rules that I had to follow. The first was not to add or subtract from the population, which growing up, I think makes a lot of sense, especially as a young girl in the South. The second was not to just cry, but to also use your words. I'm a person that feels things very emotional, like very deeply, and I'm very compassionate and empathetic. And as a child, I would often, as children do, just cry and not explain myself. And my parents were they really pressed me to make sure that I could communicate how I felt and why I felt the way that I did. The third rule still serves me well, even to this day, which was when in doubt, think, will this upset your mom? And you can do with that information what you will, but you like run that question by yourself. And then the last and most important rule that we really had was that I had to go to college, which I think a lot of people can relate to. My parents were really pressed upon me the need for a college education, partially because my mom never finished college and my dad was afforded the opportunity to go to college through the Army ROTC program. But that was really it. They didn't care what I went to college for. They didn't care what I studied. They were really just like, go and have fun and get a degree. And so given that, I've only ever had like a couple of career goals in my life. I've only ever dreamed of being a few things. One of them, probably my earliest experience in life in 1992, I wanted to be a Christmas elf. I was really dedicated to that goal. I'm a really big holiday person to this day. Unfortunately, this dream was ruined for me by a gentleman of the name of Clark Fisher, who I'll never forget, in the 2nd grade. And I like to talk about him every time I give this kind of talk. And so after that, I decided that I wanted to be a woman that has a nice office, like the way that women in romantic comedies and movies don't have jobs, they just have like really nice offices. And I was like, I can do that, I can nail that. And probably like a couple of years after I decided that I wanted a nice office, I had this science outreach program that was sponsored by the University of Georgia, where I got to dissect all of these different animal brains and sheep brains. And it was really cool. It was one of the few hands-on experiences that I had in like public school, like in my school system. And it really stuck with me. So I decided after that experience that I wanted to be a neuroscientist. And I think really after that experience, I decided I wanted to study the brain. I didn't know what a neuroscientist was, but it was important to me because it was the coolest thing I had ever done. And up until this point in my life, I was not like a science girly. I did not enjoy science. I liked reading books and talking about books. I liked listening to angsty music. And that was really it. So when I applied to colleges, all of my teachers were really surprised. They were supportive, but they were surprised that I wanted to do this neuroscience thing. And so I ended up going to Agnes Scott College, which is a private all-women liberal arts college in Georgia. And the reason was because they had a well-defined neuroscience major that had been around for some time. And in my time at Agnes, I had like numerous research experiences. I did research at Georgia State, at Vanderbilt, at Emory, and at Agnes itself. And while I was there, one of my mentors was like, you should go to graduate school. And I agreed, mainly because I didn't know how to get a real person job, but I knew I was good at school. So I said, yeah, I'll go to graduate school. And when I was in graduate school, my career goal really became just to finish graduate school. I really just was like, I got to get out of here. And whatever happens, happens. But while I was in graduate school, I applied for and earned the F99K00 for NINDS, which essentially funded the last 2 years of my graduate research and now funds 4 years of my postdoctoral fellowship. And so when I think about my career goal and as I grow up, for me, it really is about this Maya Angelou quote. And I don't have a great Maya Angelou impression like Manu, so I'm just going to read it normally. But it really is this idea that like my mission in life is not merely to survive, but really to thrive and to do so with compassion, passion, style, and humor. And I try to bring that to every space that I go to. And I think it's taken me a really long time to get to a place where I'm comfortable doing that, but I feel like I've had really great success in the research that I do, in the action that I've taken, and I'm really fortunate to be in really supportive environments. But I do think one of the big reasons that I have been so successful in my postdoc is because my mentor and I have really cultivated this idea about what it means to like be a scientist and do science, to reformat that and what it looks like. And for me, one of the guiding principles of how I do my research is this convoluted metaphor and this idea that I think fundamentally the development of a neuron is really similar to the development of a scientist. And so I'm going to walk you through that as an introduction to the research I do, but also more of an introduction to myself, right? And so to really understand this thing that I made up, you have to understand how neurons form, right? And the foundation of neural development. And so early in development, you have these common progenitor cells. And over time, these common progenitor cells will divide or proliferate in order to just increase the surface area of the embryo, right? Because you need more cells to make an organism. Eventually, these common progenitor cells will exit the cell cycle and they'll begin expressing this increase in proneural genes, and they'll become neuronal progenitors, where their sole job is now to just populate the surface of the brain and the spinal cord. These neuronal progenitors will eventually differentiate into immature neurons. And so these are neurons that are really lacking any sort of structure, right? They don't know where they're going or what they're doing. They're just like bebopping along until they get to where they're supposed to be. Before eventually these neurons maturate and they give us the diversity that we're used to seeing in cortex, right? They separate a cortical neuron from a thalamic neuron. They separate a spinal cord neuron from a visual neuron. And they have distinct morphologies and functions. And when we think about this process, what orchestrates it or what really controls neurodevelopment can be grouped into two big categories, right? The first are these extrinsic regulators, and you can think of these as like environmental signals, right? Physical contact mediated changes or secreted molecules. And the second are these intrinsic regulators, things like DNA and genes and transcription factors, these transcriptional waves. And I spent most of my graduate work focusing on these intrinsic regulators, and my postdoctoral work focuses on the extrinsic regulators. And for me, when I think about that in comparison to how we develop scientists or even how we train them, when you start your scientific interest, you have many scientific ideas and many classes that you have to take, right? Things like OChem or plant biology and cell biology. And over time, if your goal is to become a neuroscientist, you sort of specialize, right? You eventually exit this general study of science and begin increasing your neuroscience training and your neuroscience research and studies, perhaps. Over time, you'll become a student or a trainee, right? A mentee, where you too are sort of like looking for connections, trying to find your scientific niche, trying to figure out where you're going to put down roots and connections, and both in your life and in your research. And eventually you'll maturate to become a mentor, where you'll train other students and other people within your discipline or within your community to help them reach their goals as well, right? And I think it's important to point out that this mentor relationship is reciprocal, right? Like even your mentors have mentors, and it's a part of, I think it's a beautiful part of the metaphor that keeps going, right? In the same way that neurons have circuits that develop over time, mentor relationships also can do that. And when we think about the orchestration of this process, right, of developing scientists, we can use me as an example, but I think the factors are the same, right? It's these extrinsic regulators, right? So my identity as a Black Samoan queer woman growing up in the South in this multiracial family represent those intrinsic factors, who I am as a person and what I bring to every space that I walk into. But when we look at these extrinsic factors, it's really the decisions that I made, right? It's the actions that I took, my decision to attend an all-women's college, to go to an interdisciplinary program for graduate school, to apply for various fundings and earn them. But another big part about that is also the people, right? The people that influence my work, the community that I build, and the people that push me through to each step of this process. And I think for me specifically, an even bigger and probably more long-lasting effect than all of this is Black in Neuro, which is a nonprofit that was really made by 24 early career scientists in 2020, after the murders of Breonna Taylor and George Floyd and Ahmaud Arbery. We really came together to ask, how can we best create a sense of belonging and support for Black neuroscientists within the community? And so much of this success has been due to social media, has been due to the forward change in donations, but also just the participation of other Black neuroscientists. And I actually remember when we had the first spin week in 2020, we had a Black in Neuro roll call day where we all just like introduced ourselves on the internet. And I remember I actually got really mad because I was like, where have all of you been? Like, where have we been from one another? And so it's been an honor to sort of build this institution and this nonprofit that's gone out of its way to really pay its speakers, provide resources, and start conversations and discussions throughout all of that. And I think one of the reasons that all of this is so near and dear to me is because I see it as a foundation for how I view research, right? And so hopefully through that metaphor, what you can appreciate on some level is this idea that I'm really interested in understanding how both trainees develop in and outside of lab, but also how cells develop in normal and pathological conditions, right? To me, those two things are parallel to one another, and you can use the principles and the concepts of one to infer the other. So when we talk about trainees, the questions that I'm interested in are really like, what are the systemic principles that deter historically excluded students from pursuing careers in neuroscience? How can we use the principles of neuroscience, like that metaphor that we talked about in the last couple of slides, to generate and maintain a sense of belonging for students? And I think a question that any like PI or faculty member wants to know the answer to, how do we train and support successful and happy researchers, or neuroscientists in my case? And hopefully what you can appreciate is that the questions that I'm interested in asking and the research component to that are parallel to these, not just because I wrote them that way, but because they actually like make sense, right? So why do rare genetic diseases predominantly affect the nervous system of children? How do the pathogenic mechanisms disrupt cellular and molecular events during neuron generation and development? And just simply, how are mature and unique neurons generated and maintained? And for me, these two ideas really have to go hand in hand because they show how much you can appreciate both an individual, like an individual person, but also an individual cell. And it reminds me of this really great quote from Bell Hooks that talks about building and forming beloved community, right? It's not about the eradication of differences, but really the celebration and the claiming of your identities and cultural legacies, that every piece of you makes you who you are and contributes to the community that you build. And I think that that's true both for the brain and for people. And so when I think about systems biology, it's really the best approach to do a lot of this research, because in its nature and in its definition, it really is all about putting pieces together, whether that's understanding the genes that are mutated that informs molecular networks that we look at, whether that's looking at those molecular networks to then investigate cellular pathways and circuits, and then making informed decisions about how the brain responds to a disorder and how the individual and personalized medicine, can take that a step further. And one of the things that I love so much about doing systems biology is that I get to do it in the Fondas lab, which has been a dream position for me and has really pushed me to think about science in a different way, where we use these principles of molecular systems, where we use the principle of molecular systems biology, biochemistry, function, as well as genetic approaches to ask really fundamental questions about basic diseases, but also what occurs at the synapse. And so my project focuses on this idea of these extrinsic regulators that I brought up earlier, right? And so these extrinsic regulators really play a role in things like cell migration, where during cortical development, as early as EE 14.5, you have these neurons that will scaffold up radioglia, and until they come into contact with a molecule called renin, they'll keep going, but eventually they'll generate the cortical plate. So extrinsic regulators control this propagation. You also have extrinsic regulators being important for things like mitochondria integrity and mitochondrial health, as well as overall cell homeostasis, right? How does calcium work? How does calcium regulate cells at the synaptic level for me? And then I think most common within the brain, these extrinsic regulators are really studied in terms of function. So how are synapses built? How do neurons talk to one another? And how do they propagate the action potential over time? And so our lab really focuses and studies copper and its role in the brain. And what I really want you to gather from this slide is that copper is important for the brain. And this is a fact I did not know until 3 months before I joined the lab. So if you're just finding this out today, please don't feel bad. But copper really acts as a cofactor in a lot of cellular processes. And because it's expressed in these sort of small concentrations, it sort of gets disregarded, or disregarded is probably the best word for that. But it plays roles in things like neurotransmitter synthesis. It plays roles in mitochondria health and maintenance. And it's been associated and linked to several neurodegenerative diseases, including Alzheimer's and Parkinson's, Wilson's disease, and Menkes disease, which is the disease that I'll talk about today. And so when we think about Menkes disease, it's a rare form of childhood neurodegeneration. That symptoms really include this kinky hair, loss of pigmentation, as well as loss of collagen. A lot of the patient's symptoms include things like intellectual disability and failure to thrive. And most patients that have Menkes disease don't live past the age of 5. On a cellular level, when we think of Menkes disease, we're talking about gross cell loss and brain atrophy, where Menkes disease predominantly affects cerebellar Purkinje cells, which have these beautiful elaborations and dendritic arbors. But in Menkes disease, they're sort of shrunken and shriveled, and they never reach that level of complexity that we're used to seeing over time. And so one of the interesting things also about Menkes disease is that we know exactly what causes it. It's caused by mutations in this copper transporter ATP7A. Now, what hopefully you can appreciate from this beautiful cell schematic made by Courtney like 3 years ago is that when copper enters the cell, it does so through this transporter called CTR1. And once it gets in there, it does a whole bunch of things, right? It's playing roles with the mitochondria, it's being degraded by SOD, but it also has to leave the cell. And to do that, it does so and is shuttled through via ATP7A. Via ATP7A or ATP7B. And Menkes disease is a global depletion of copper or systemic depletion of copper from the body, not just the brain. And so these effects are really deleterious, is the word that my PI always uses. And so one of the first challenges of the lab was really to map the ATP7A interactome. So if we know that ATP7A is the reason that Menkes disease occurs, what are the other molecules or genes associated with those deficits? And so we create these interactome maps, which I'll show you one in a second, but it really does kind of look like this. And from there, we're able to look at specific nodes and isolate genes or proteins and perform candidate testing where we mutate or overexpress genes either in fruit flies or in human cell lines. And then we can see if we can recapitulate or redefine these Menkes phenotypes. Can we generate Can we generate neurons that look like our Menkes phenotype? And can we rescue those functions given what we know about the interactome as a whole? And so what we see, or what the lab saw really when they first did this in 2017, was that the ATP7A interactome in and of itself is enriched in genes that are involved in both neurodevelopmental and neurodegenerative disorders, which was really a pioneering feat at the time and was one of the first times that Menkes disease had been studied in this sort of global perspective or global way. And one of the sort of interesting genes that was isolated from this study was the COG complex and its role in upregulation in protein in cells from patients as well as their age-matched controls. And I think what's interesting here, and what was really cool for me actually, because I studied the COG complex in undergrad, which feels like so long ago, is that the COG complex is actually incredibly important for Golgi cargo retention, but most of the symptoms associated with deficits in the COG complex are the symptoms of Menkes disease on a cellular level. And so we wanted to investigate that further. But what the COG complex does is essentially tether vesicles to the Golgi apparatus so that it can be so that they can be processed. And so within a cell, this is really what it looks like, where the COG complex is this sort of crisscross hair tie, if you want to call it that allows vesicles to move throughout the cell and then eventually end up in the ER or degraded into lysosomes. And like I said, deficits in the COG complex or mutations in it are associated with things like brain atrophy, hypotonia, intellectual disability, and epilepsy, which are all very common symptoms associated with Menkes patients. And so we really wanted to understand how these COG null cells work and if they have any challenges or changes in copper within them. And so we took human COG null cells we took human COG null cells and we performed immunoblot first to look at the expression of ATP7A. And hopefully what you can appreciate is that in these cells, there's a decrease in this copper transporter, so the thing that removes copper from the cell. We also wanted to see how the cells work or respond to a copper challenge. So we treated the cells with different concentrations of copper, as well as BCS, which is a copper chelator or a negative control in this case. And what we see here is that these cells have a really hard time absorbing metals, suggesting that CTR1, the way that copper gets into the cell, is also affected. And so what this creates, obviously, is this idea that these human COG null cells are ATP7A and copper deficient, and perhaps there's some connection between the way that COG works with ATP7A. And so the strategy to test this was really cool, and I was really excited about it. It was using the Drosophila neuromuscular junction in the GAL4 driver system. And for the sake of simplicity, the GAL4 driver system really allows us to express human genes in flies. And the GAL4 part of it allows us to do it in a cell autonomous and specific manner. So we can target just sensory neurons or just motor neurons. And a lot of the work I'm going to show you is in collaboration with Dr. Dan Cox at Georgia State, who is one of the most patient men I've ever met in my life, as he has taught me about dissection and Drosophila husbandry and all kinds of fly behaviors. And so I'll definitely point out which experiments we did together. But so for us, it was really trying to see if we can establish that ATP7A and COG interact and have a difference in an impactful way within the Menkes synapse. What we found when we did the first round of these experiments, which was before I joined the lab, but you can learn more about it at this QR code here, was essentially that there are morphological changes in the synapse when we have our ATP7A mutation, which is recapitulating Menkes disease, and our COG mutation. But combined together, we're able to rescue that function, right? So, and these are synapses that have been stained in green with a GFP, and C155 is a neuron-specific driver. And the length of the synapse has been quantified for you over here. But really the takeaway is that the Menkes flies, or flies that have our Menkes phenotype, have this decrease in overall synaptic length. And when we combine that with our COG1 mutation, we're able to rescue on some level these changes or this decrease in morphology. And we also see that mutations in COG1 affect the synapse in and of itself. We also looked at the functional aspect of this using neuron evoked potentials. And essentially what I really want you to gather from this is that we're seeing this morphological change at the synapse, but there's also a functional consequence to it, where our Menkes flies and our Menkes flies are having this sort of increased amplitude of expression and firing, but we can rescue that when we combine that ATP7 mutation with COG as well. And so what I think this really points to, and what the lab really wanted to know, was like, what are the cellular factors contributing to these synaptic changes? It's obviously more than just us mutating two genes that are kind of next to each other. And the answer really comes in the form of mitochondria, because mitochondria are dynamic. And so they're constantly moving from the cell body to the synapse. And so they play a really big role in things like calcium homeostasis at the synapse, as well as cell signaling. And this is where my part of the project picks up. And this is me on the day of my defense. And so what we see when we look and when we use MitoGFP to look at the mitochondria content of these Menke synapses, essentially, is that they're devoid of mitochondria, which is so weird. And I remember when I saw this data, I thought I just did it wrong. But what you have here is the neuron plasma membrane in magenta and the mitochondria in yellow. And in our ATP7A overexpression, which is our Menke's phenotype, what hopefully you can appreciate is that this is just like black. There's like no mitochondria within the synapse. We're able to rescue this phenotype if we also have mutations in the COG complex, but it's pretty odd. And these are in motor neurons. We see a similar thing as well when we look in sensory neuron dendrites, which is the work that I did with Dan to really map out where, and this is quantified for you over here, but in comparison to our control, our Menke's phenotypes and synapses have less organelles per like cubic pixel, but yeah, and that we can rescue that to some extent. But the question is really like, what is going on? Like, where are all the vital things that make a cell a cell? And the answer is that all of the mitochondria are stuck in the soma. So this is the ventral nerve cord of the Drosophila, which really is the cell bodies. And what hopefully you can appreciate from this yellow staining is that these are all the mitochondria that should be in the synapse. And we see very little in these other, in our other fly mutants. And so I like to think of this as the idea that the mitochondria are stuck in traffic. And I live in Atlanta, so I hate traffic 100%, which makes complete sense since the state is like run by traffic. But if you think about it this way, the idea of where are the mitochondria, they're stuck in traffic. This is like a real picture of Atlanta traffic. And I like to think of that as a bunch of cars being stuck on the highway. And one of the first questions that I always ask as I'm screeching to a halt in traffic is like, what is going on? And there are always like one or two options. The first option is that there's damage to the literal road, right? This is the unfortunate 75-85 bridge collapse in Atlanta, but that there's issue with the literal road. We know that mitochondria move through the cell using microtubules and kinesin and dynesin. So is it because there's something wrong with the motor proteins that the mitochondria can't get from the soma to the synapse? The second option is that there are too many accidents. There are, like, if all of us are on the road and this side of the room is like, has to call hero because they all got flat tires, that's where nobody's going anywhere until that gets resolved, right? And so that's the second possibility, right? For these mitochondria, are there so many damaged mitochondria within the cell that it doesn't have the resources to respond and get rid of all of them? And this really focuses on the idea of mitophagy. And so we decided to look at option 2, one, in an overly honest methods way because we had the flies, but 2, because it also was like the biggest bang for our buck because it would be really cool. And so what we did was we took our, Menke's flies, and we also we added in mutations to either PARC or pink, which are 2 components of the mitophagy pathway. And what hopefully you can appreciate is that by inhibiting PARCin and pink within the fly, we're able to rescue the synapse morphology. Like, we're able to allow the synapse to re-elaborate. It is definitely like an over rescue. So they're definitely way more elaborate than controls, but it's really interesting. And it's kind of counterintuitive if you think about it, right? When we talk about mitophagy, and this is about to be like the simplest explanation of mitophagy ever, what it really is that damaged mitochondria send out these signals, and pink and PARCin come and they tag them, and then they get degraded in the lysosome. And so what we're saying essentially is that it's better to have tons of damaged mitochondria around for synapse structure than it is to try and get rid of all of them, to get rid of all of the damaged ones. And so this project is still ongoing, but we're still piecing together a lot of the data. And one of the cool things that I get to show you is a study that was done in collaboration with me and another member of the lab, Amanda Friedman, where we tried to see if Parkinson genes outside of Pink and Park were also associated with ATP7A, or if they could rescue survival. So these are flies that have been given a copper challenge for 3 days, and then co-expression or deletion with various Parkinson genes. And we see that we're able to adjust and we're able to rescue mortality for a lot of these flies by mutating these Parkinson genes, suggesting that on a different level, there's a connection in the neurodegeneration between ATP7, between Menkes disease and Parkinson's. And so we're piecing the story together, trying to understand why the Menkes synapse is void of mitochondria. But what we have shown is that ATP7 and COG interact to sustain synaptic transmission as well as the mitochondria pool. We also emphasize and are really trying to look at what role does Pink and Park play within this process. And I think more importantly, since are we redistributing the mitochondria within this sector, and what are the functional consequences of us essentially like removing mitophagy from a cell? And then lastly, a big part of my project that I'm also going to try and breeze through is the role that mitochondria play in neurodevelopmental disorders in general. And this is a paper that was done in collaboration with me and some of the grad students in the lab that was published what feels like recently, but was in 2021. But it's really looking at the idea of mitochondria diversity as a tool to study other neurodevelopmental disorders. And so we really wanted to know if the nuclear proteins in the mitochondria differ across brain region and cell type, and if we could pierce that out using proteomics, RNA-seq, and single cell RNA sequencing as well. And so we look at the proteomic data. What hopefully you can appreciate from these dot plots is that when we compare cortex and hippocampus, cortex and striatum, or hippocampus and striatum, there's increased expression of very specific nuclear encoded genes within the mitochondria between brain regions. We also see this on an RNA sequencing level, right? When we look at whole cortex versus specific mitochondria upregulated genes, we're able to see enrichment in specific enrichment in genes associated with different mitochondrial processes and biosynthesis. And we're also able to use TSNE to really to sort of drive home the point that we can identify these cells by brain region, but we can also do it by neurotransmitter. So we can sort of create an atlas or a map of the way that the mitochondria are expressed throughout the cell on an RNA level. And this is what that looks like, where the green really here represents the neurons that are GABAergic, and you can see their expression throughout different parts of cortex. And one of the reasons that I love talking about this paper, even though I just really breezed through it, is this idea. It gives forth this idea that systems biology is a tool. It allows us to think about science in a nitty-gritty way and piece it all back together. And it reminds me a lot of this quote by W.E.B. Du Bois, this idea that education must not just teach work, it also has to teach life. And I think that there's a lot that can be learned from the connection of mitochondria and the small role that they play within the brain, but also, the complexity of the people who do neurodevelopmental research as well. And so we do this in the lab, obviously, by looking at mitochondria and looking at their diversity and heterogeneity. But for me, it really underscores the idea that the principles of diversity, equity, and inclusion are a fundamental part of neuroscience research in the way that the brain functions. But we also do this by writing commentaries and pieces about the impact of Black neuroscientists and Black mentors on neuroscientists. And also holding ourselves accountable while we try to build community to reflect on the things that have and have not happened. And with that, I just want to thank my lab. Obviously, I want to thank all of you for listening to me talk for hopefully the appropriate amount of time. And yeah, if there are any questions, I'll be happy to take them.
Okay, thank you for your presentation. It was really nice. And I'm pretty sure there's a lot of information in those papers that you couldn't summarize. And I have two questions for you. I didn't think that the hypothesis of the stuck mitochondria was viable because you should have been able to see the mitochondria on your immunofluorescence, right? I mean, you didn't see them. So that means that somehow they were either damaged or disappear. If they were stuck in one place, you will see them in your immunofluorescence. So that's one thing. The second one is your approach of having damaged mitochondria might be better than having no mitochondria. And I'm going to toss something for debate with you. And I'm not sure that is good, because you have damaged mitochondria, you have more radical, free radical species there that creates more oxidative stress, more damage in DNA, and a lot of more potential degenerative disorders or even cancer in some places. So I would like to pick your brain and what do you think about this? If you can comment.
Yeah, that has been actually, I'll answer your questions, I guess, in the reverse that you asked them. But that has actually been one of the most confounding things about doing this research, is the idea that like damaged mitochondria are helpful for the synapse and on some level appear to be helpful for fly mortality. And I think what we're missing, and what we're also trying to look at and investigate using mass spec and sort of big proteomics data, is the viability and the health of the cells themselves. So just because a fly can live for a couple of days, does it mean it's very happy? Does it mean there aren't functional or structural changes? And so one of the things that I'm trying to investigate is like, how do damaged mitochondria impact cells? And you mentioned like a bunch of very great ways, but also what are the consequences of that? And is there a way for me to like viably see that within the fly, or if it's better to move to like cell lines or cell models? So I would absolutely love to talk about that because I think it's really cool. And then the first part about the mitochondria sort of being stuck in the soma, or if they're damaged. We see, if we image like the whole of the synapse, neuron cell, there you go. If we image like the whole of the muscle, we have other images where we can see them sort of stuck in the soma with them devoid. So essentially like a more blown out version of that picture that I showed you. But I do think that the metaphor that I gave of them being like stuck in traffic and stuck on the highway is definitely an oversimplification of what is going on. So some of them, it does on some level make no sense that they would be stuck in the highway or stuck in the soma without a way to get out. So yeah, that is something also I would love to talk about. Yeah. Feels weird to have you raise your hand. You invited me here.
So Kayla, thank you. Nice talk. I couldn't help but notice Bada Sabag's picture in your pictures up there. That's great. I want to compliment you, if I may, on, I think your work demonstrates something that in neuroscience in particular is something we hold up, how you went after something specific in Menke and you're looking at this particular defect, but it's opened up a window into other things, neurodevelopmental disorders in terms of the role of mitochondria. I think it opens up a lot of avenues specifically. I wanted to ask you or suggest to you about, particularly in light of some of the hypotheses discussed about damaged mitochondria and density of mitochondria at the synapse, and about the dopaminergic neurons with respect to Parkinson's, whether you or somebody else is doing a neurophysiological assessment of the ability of those synapses to maintain high levels of activity and synaptic transmission. So getting beyond the molecular signatures to the kind of phenotypic readout of the function of the synapse, it seems to me there could be some really profound effects. And certainly dopaminergic neurons, where there's great ways to sense the dopamine release as well as transmission, and they fire at high rates. There's a lot of cool things to do there. I'm just curious if you're doing that.
Yeah, so we have a collaboration at Georgia State, but also at Emory with Dr. Katz, where they do a lot of the electrophysiology associated with these papers. And one of the things that they see when we look at like, what is it, tympanic neuron transmission. So essentially you stimulate a nerve cell for a really, really long time. And you then see how long it takes for it to like release vesicles. And they do see increases in that tympanic synaptic transmission, suggesting that there's an issue with plasticity, with short-term plasticity. But we're also trying to look at different cell types and sort of different physiological aspects of it. My project does not focus on that. I do make the flies for that though. And so I'm trying to learn and get more immersed in that culture. But I like this work because it's very interdisciplinary and it lends to a lot of collaborations. So, yeah.
Is it not on? Is it on? Flip it up.
Sounds like it's on.
It's green.
Feels
Green?
like it's on.
Yeah.
Yeah.
You tricked me. Need some challenges everywhere. First of all, fantastic talk, like from top to bottom. I mean, oh my gosh. So I wanted to ask two questions, one related to the science and one to the first part of your talk. But so I'm a vascular biologist, so I'm probably going to ask some very naive questions right here. But in terms of Menkes disease, I get caught in sometimes talking, having things being so, because I sit next to a lot of neuroscientists who want to make it all about neurons,
Yeah.
but right next to neurons are blood vessels.
Yeah.
And so I wonder about Menkes disease, because you said sort of it's a loss of collagen. I feel like there's got to be this synergism between synaptic firing and blood vessel dilation and nourishment of those neurons.
Yeah, that is.
So I wonder, so then in Menkes disease, is there any sort of association with a vascular component, or is it all neurodegenerative?
So yes, there is. And one of the graduate students in our lab actually is studying that. She's looking at vascularization in astrocyte in feet to try and piece that together more. She actually gave lab meeting yesterday and I wasn't there, so I can't give you an update on what she found, but it's been really cool because I, as a neuroscientist, also make everything about just the brain. And so she really pioneered that project, like focusing on the vascularization and what that could mean. So I can keep you posted.
Yeah, please do. Because I saw your sad looking neurons and I was like, wait, at least you got neurons.
Neurons, yeah.
So something else is not supporting those neurons. And I'm sure it's all the beautiful molecular mechanisms that you just described that, are related to the mitochondria. I know, don't we all, right? So then a part of that question is actually, because I loved your analogy about, sort of the trainees and cells kind of having some parallels there, and the nourishment of those cells and nourishment of trainees. Because I think about that, kind of in the terms of like your career trajectory, there being something about, as not just as maybe a scientific or an academic community, but as society at large, where cells and trainees are welcome, and the communities that are predominant in those areas providing a welcoming environment. Because I think about, the great work in Atlanta that, you and others have done and Manu described, like of going out in the community and saying, you are welcome here. Like there should be no reason you're not. But I guess maybe a comment on, being a white male, how is it that we can help structure and go out into society and help say, not just are you welcome here in the scientific community, you're welcome in our society, right? Because I think Juneteenth is a celebration of emancipation, but into now a mixed population of society, where I thought there was an interesting comment about how slavery kind of got morphed, and how that got morphed was that society sort of said, you're still not welcome, you're free, but are you welcome here is still a question, right? And I just wonder if there's a voice that, as a white male, we can have out in society that's saying, not just are you welcome here in the academic community, which we're, trying to do, but also out in society. Your talk about your career trajectory caught me on that one, because it felt like you got that vibe early that I could go in and do anything, but I wonder if there's still like a conversation at play there.
Yeah, so I definitely think there is. I think there's more than one way to approach the idea of like, how do you promote, how do you like show welcomeness, or like promote an accepting, inclusive environment. I feel like, so the one way that I always describe it to students, unfortunately, is that like it's a vibe. Like it is as a person like experiencing it, you can understand when you walk into a room if it feels comfortable or not. But then the other thing that I always try to tell my students, and a thing that I think is really taken away from people, specifically white people, in conversations that they have about their identity and their intersectionalities, is recognizing the power that you hold whenever you're in any space, whenever you walk into any room. And it's something, teaching at an all-women's college as an adjunct, I make sure that my students know and understand that like every time I walk in a room, I hold power in that room in some regard. It could be more, it could be less. But the way that I use that power and sort of speak in that autonomy is really important to define the community, right? Like I joke around with my students. I'm obviously like a very goofy person sometimes. And that puts them at ease, but I set the tone and I set the power dynamics for how that goes. And so I feel like when you're talking about building community, like across the diaspora of Black people, but also with different ethnic groups and intersectionalities, you have to make sure that you're giving people the space and freedom to be themselves. And that you're not suffocating them in that, and that somehow through your actions, there is this like, I don't know, it's like this calm and this acceptance. I think part of that could be like standing up for your colleagues in certain situations. I think part of that is also having difficult conversations and educating yourself in new ways. I don't think that there's one like defined way to do it, but I think for me, a lot of the times it boils down to having conversations about power and having conversations about what comfortability looks like and what it is. And I do feel really fortunate that like my parents instilled in me like a vibe check meter that works pretty well when I'm in any space. But I also think a lot of that has to do with me growing up as a Black woman in the South, being hyper aware of my surroundings and hyper aware of the people I'm with. And just taking that sort of like fight or flight autonomic nervous system response and being like, I don't like it here, so I'm gonna go.
Well, what I loved in there is it's kind of wrapped up in your analogy of the extrinsic factors for cells. What makes cells flourish and differentiate into the beautiful heterogeneity that emerges in the brain? You have to have like a supportive environment. Otherwise, those progenitors will kind of shrivel up and not form the right synaptic connections, right?
Yeah.
So that's why I love your talk so much. So thank you so much.
So we have time for maybe one more question if anybody else has a question.
Yeah.
Do these mics? Are these? Okay.
So when you created the group Black in Neuroscience, how did you find members? I mean, you can create these groups, but how do you disseminate and how do you find members?
So we really just use social media to find members. We use Twitter. It was also right after, I should give more context to the founding of Black in Neuro. It was after Black Birders Week and Black in the Astro, and Angeline Dukes, our current president, she just tweeted like, when are we gonna have a Black in Neuro week? Because it was beginning, people were like using hashtags and starting to form like Black in Physio Week or Black in Cardiology Week. And so people essentially responded to that tweet asking to like help with the organizing. And we literally, she sent that tweet on like a Monday. By Saturday, we had like a Zoom meeting to talk about like how we were gonna plan and schedule our week. And then we just like super promoted it on social media. We talked to our department and like all of those weekly spam newsletter emails that everybody gets. I get like at least 7 different ones, but we sent sort of announcements about Black in Neuro Week to those people. We asked people for donations and for sponsorships. And so it was really by word of mouth, but it was also a lot of like legwork by the like trainees and the different organizers. And I think now all of the other Black and X communities have been super supportive of one another. And so it's really by word of mouth and by like everyone introducing everyone to their networks and their sponsors and things like that, that we've been able to sort of like grow that community. So right now our directory is, I think our membership directory is like 500 plus Black neuroscientists, but not just neuroscientists, like people interested in neuro related fields in general. So that's PhDs, MDs, PsyDs, PharmDs, and it's also searchable. So if you're looking for like people to invite for seminar series, whether that be virtual or in person, it's searchable by career stage, it's searchable by intersectionalities, by if they're looking for jobs or not, and also by research topic. Yeah.
Well, thank you, Kayla, for that talk. Okay, so now we will have like a short 15-minute break and we'll meet back here in this room at 10 o'clock for our next speaker, who will be Dr. Margie Lee.
Thank you. Now, just a few words. I want to welcome all of you visiting here to the Star City, Roanoke, 7-time All-American City. And we're pleased that you're here. I want to thank Dr. Fralin and his work here and putting this together. This is a great thing. And let me just tell you, I'm a very passionate mayor about treating people well when they come to my city. So if anybody gets any citations or things or stopped by the police, call the mayor's office. And I guarantee you within 40 minutes, you're gonna call back from my office telling you where to go to pay for it. So that's a compassionate mayor. I want you to ensure, I don't want you to have to figure out where to go. We'll take care of that. Thank you so much. Thank you.
All right, welcome back, everyone. My name is Kenneth Young. I'm a fourth-year MD PhD candidate at Virginia Tech Korean School of Medicine and Fralin Biomedical Research Institute, and I'm in Dr. Samuel M. Wee and Dr. James Smith's labs. I will be introducing Dr. Margie Lee, who earned her bachelor's in biology with a minor in chemistry in 1982, and her doctor of veterinary medicine degree in 1986, becoming one of the first Black graduates to do so in the college's veterinary program alongside her classmate, Dr. Lynn Hoban. She went on to complete her master's and doctoral degrees in medical microbiology from the University of Georgia in 1988 and 1990, respectively. As a veterinarian and medical microbiologist, Dr. Lee practiced in veterinary medicine in a private practice and conducted postdoctoral research at WashU in St. Louis before joining the University of Georgia faculty in 1992. While in Athens, Georgia, she served as a diagnostic laboratory director of the Poultry Diagnostic and Research Center and held a joint appointment as a professor in the departments of population health and infectious diseases in the College of Veterinary Medicine. Dr. Lee is the current professor and head of the Department of Biomedical Sciences at the Pathobiology in the Maryland College of Veterinary Medicine at Virginia Tech, the first Black American and woman to hold this title in the department's history. Dr. Lee's research is focused on the epidemiology of meat and poultry-borne food safety pathogens, ecology and mechanisms of antimicrobial resistance, and ecology of poultry intestinal microbiome, and has been featured in 77 peer-reviewed publications and holds 2 US patents. Dr. Lee is a highly accomplished scholar, researcher, and leader. It is my distinct pleasure to introduce the 3-time Hokie, Dr. Marjorie Lee.
Yeah, where do we control slides? Thank you. Okay, I think I understand this now. So thank you very much for inviting me. And so I feel like a little bit, I'm the polka dotted unicorns among the unicorns. And I say that partly because I'm probably maybe the only boomer in this group. And so I'm going to give a boomer talk. So get yourself prepared. It is what it is. The other part that makes me the polka dotted unicorn is that I'm probably the only local girl. So how many of you guys speaking today are native Virginians? Check it out. How many people in here are native Virginians? Okay, there we go. So I grew up about 35 miles from here in Bedford County, Virginia. And most of you guys know about Bedford County. It's, it's got Smith Mountain Lake. It's become the really wealthy retirement area, right? Well, I'm not from that part of Bedford County. I'm down the country part, down near Campbell County, which there is no lake, sadly. But let me go back. Here we go. One of the things that, I want to talk about as a boomer talk, I think, is how much things have changed. Since I've been in science, and how much things have changed since I've been really here. So I was born in 1960, and the 60s were a hot mess. You guys think things are bad. They were shooting presidents in the 60s. And anybody who thought they wanted to be a president, and anybody who thought they wanted to be anybody. So there was a lot of shooting going on in the 60s. And what white people saw was the decade of love and drugs and birth control pills, and they were free. But, you know, there were church bombings going on, little children dying. I mean, what you guys are seeing with the school shootings was more of a political movement, I would say, in the 60s in an effort to keep people in their place. And the development of opportunities in America, the land of opportunity, has been slow for some, but certainly the progress has been sustained. And I show this picture here. I started doing genealogy, and one thing about in the South, and especially in Virginia, which likes to document everything, there's a genealogy museum in Bedford County, and I found things about my family. I found a whole written history from the oral history of the family that I hadn't even heard, written up in a document in the Bedford County Genealogy Museum, where one of the first Black schools in my county was actually built by and the land donated by my grandmother's grandfather. And so what I did not know was that I was marked probably from this profession from a very early time, that education was valued, opportunities were not great. But it's America, people make their own opportunities in a lot of cases. So this school, Johnson Mountain School, which was documented there, was one of the first Black schools built by my great-great-grandfather, and was still an active school in the 60s until they integrated. And so I had the advantages, I think, in some degree of being able to go to integrated schools all my life, and made the decision early on in going to college that I would go to a white college, because up until that point, we couldn't. And I made that my goal, but then things changed a bit. So my family wanted me to be a teacher. That's what my grandmother always told me. She said, I was in training to be a teacher. My father died from a heart attack. I couldn't finish school. You'll be a teacher. Well, I didn't want to be a teacher. I wanted to be a veterinarian because I love horses and animals. I didn't like people much. I was fairly certain I could escape that whole thing by becoming a veterinarian. I was oh so wrong. And so, of course, medicine means science. And I knew that I had to concentrate on those things in school. But I come from an area where work ethic, it was everything. Frankly, still is everything. And putting in the work is going to be part of what I'm going to talk about here in building credentials and building a reputation in science. And so part of this, I'm going to be speaking sort of as a historical basis, I guess, as a Black woman in science. And the other part is how to build a reputation that's sustainable. So you see this part. I thought I was the first in my family to get a bachelor's degree. And then I found out the West Virginia cousins beat us out. So anytime you hear people like bad-mouthing West Virginia, and then of course I go to vet school. There were 2 Black people on that part of campus, me and Lynn. There were no Black faculty. There was no Black staff. Even the janitor was white. It was quite an experience, but that experience was normal for us, because when I went to Virginia Tech, and we actually looked up university statistics. The statistics said that there were 100 Black students at Virginia Tech, but we could only find 100. We all knew each other on a campus of nearly 20,000 students. And so coming up in science, and you guys are probably still experiencing this, I think, to a degree, where in a lot of cases, you'll be the only brown face in the room. And being able to navigate that from a professional standpoint, but also from a reputational standpoint, is really important in the first part of your career. And so, in speaking about that, I'm going to tell you a quick little story. So I go to the University of Georgia as an assistant professor. To my knowledge, there were 3 of us. And I'm going to say Black a lot, honestly, because in my lifetime, we were, let's see, the N-word was definitely very active. We heard it a lot, but we were Negro, colored. Black was when we determined our own autonomous identity. And then your generation, African-American, and I'm sure there'll be another in my lifetime. So I go to the University of Georgia as an assistant professor. To my knowledge, there were only 3 of us Black faculty in STEM, and we were all assistant professors. And we got stuck in a lot of recruiting, what was beginning to be diversity efforts. And the belief was that they had a really hard time finding Black students that had good grades and good test scores. And that was the belief. We have a hard time having a class that have a suitable number of Black students that are qualified. And so I'm on this committee where we're looking for students for an undergraduate research program, a summer research program. And as part of the application, they'd put their GPA, they'd put their strongest science subjects, and they'd also put their SAT and their PSAT scores. And they're getting PSAT scores that are really high for these Black students. But then the next line says, where do you intend to go to college? Well, it wasn't the University of Georgia. It was Morehouse, it was the Ivies, it was the top 10 and 20 in the rankings, but it wasn't the University of Georgia. And so what I thought then, I was like, there's this nice size pool of Black students with the scores and with the grades that are getting the experience and they're not coming here. Science might really look different in a decade or two. What you're seeing here today, I'm misting up a little bit. I'm not gonna lie, it's a little emotional. I'm seeing the results of that, where these people went to Morehouse, they went to Harvard, they went to Yale, they went to Stanford. They're in positions now of influence because they've made their reputation. But a lot of the universities weren't able to recruit those that didn't know. So now you're seeing, so the question that I asked her about, how did you recruit? For us, we could have social media'd all we wanted. There was no fish in the pond to catch. So that was, for me, the really good part of this program. I don't think I've been this moved since I went to the first majority Black college, was Alabama State. I was invited to give a talk. And so I roll into campus, I park. I had to sit on a wall there and try not to have a heart attack because it was a campus full of Black students. I'd only been at white universities. I'd never seen that before. And so now seeing so many Black scientists in the field. So I get through vet school. I go to vet school to be a horse vet. You saw me on the pony. I wanted to be a track vet, actually. About halfway through vet school, I realized that was not going to work for me because number one, I'm kind of a no drama zone when it comes to work. I'm a left brain. I'm analytical. I'm a spreadsheet medicine person. You can't do that with horses. It's not spreadsheet. And I knew that equine medicine wasn't going to work because I was annoying my professors by asking questions about things nobody knew the answer to. I was like, well, why do we do it this way? And how does that physiological pathway work? And can't we intervene with this? And they're just looking at me. My first semester, we had microbiology, I think, no, maybe the second semester. And there was a question there about serological tests for detecting this disease in cattle. And it was a discussion question, and I answered the question. The professor stopped me in the hall and said, "You're the only one in the class that got that right." It's like, which question? That question? That question was easy. And I started rethinking how I was gonna use this veterinary degree, which is why I had to rethink to find analytical parts in medicine that wasn't just single patient treatment, but either populations or mechanistic or analytical. And so I decided to go get a PhD. I went to the University of Georgia to do it because they had a pretty well-developed program in infectious diseases. And I got a PhD in bacteriology. And so I will say this to all of you young trainees in the room. You will have these characteristics that you think are personality flaws, and I can tell you there's a profession for that because I have always, and my mother is in this room and will testify, I have always been interested in pus, snot, and diarrhea. Who knew you could make a living at this? You could get hundreds of thousands of dollars to do research in pus, snot, and diarrhea. I mean, who knew? I had a boyfriend once, he was from Campbell County. They had acne. I found that attractive. So this organism, Pasteurella multacida, which was named after Louis Pasteur, who actually developed, this was really the second renaissance of microbiology, Louis Pasteur working with this bug, and its superpower is pus. It also kills things really quick, especially turkeys. You can infect turkeys today, they'll all be dead in 36 hours. I mean, just, it's the cholera of turkeys. And so I got my PhD in a bacterium that had a lot of interesting characteristics and no real clear method of pathogenesis. And method of pathogenesis means, how does it do this? It didn't have any toxins. It didn't make any interesting enzymes. The only thing it could really do well was grow in blood. And you almost can't find an organism that can grow in fresh blood faster than Pasteurella multacida. I mean, it'll grow in blood faster than the lab. How many of you guys have had that cat or dog bite where your hand swells up real big and they have to lance it and drain out all the pus? I know a couple of you have. That's Pasteurella multacida. So a cat bite, like in the morning, my husband messed with the cat one day and a cat bit him in the morning. His hand was swollen up by noon. That's multacida. And so what was fascinating about it, when I was a graduate student, I started looking for toxins and enzymes and only made one extracellular enzyme activity, only one. And I remember writing in my lab notebook one day, I need to study this enzyme. But there were a lot of barriers to it, which I'll tell you about in a minute. And so after I got my PhD, so right now I got 4 degrees. I've been in college for 12 years. My grandmother said to me, when are you going to come home and open a practice? And I said, I don't think I am, Grandma. And she said, well, when are you going to get a job? And I'm like, soon, Grandma. And so I've got faculty offers. I have the credentials for a faculty position. The economy was such that universities were expanding. It's just one problem. You can get the position, can you keep the position? So what are the metrics that you're going to need? What are the resources you're going to need to be able to compete at the highest level, which is what it is to get grant funding? And so I use sports analogies for it. You know, not many people can come out of high school and compete in the NBA. And it's like that for research. Not many people can come out of a PhD and compete at the highest levels for research funding. And I knew for sure that I could not. And I turned down the faculty positions and did a postdoc because there are a couple of things I needed. Number one, in order to be able to study these organisms, I needed to understand how to use a genetic system. And there was no genetic system for my organism. I was going to have to create one. And so I went and postdoced in a lab that worked with E. coli and salmonella that had genetic systems. You got to know how to clone genes. You got to know which approaches may work. You've got to know how to make knockouts. And then the third thing that salmonella people had, dang it, was they could move genes from strain to strain. And that's tough to do with most organisms. I thought I might be able to do it, but I knew I'd have to develop those things with my organisms. I was very good with animal systems. I was very good with physiology. I was a good microbiologist. But I needed these other things in the toolbox to have the whole picture for competitiveness. So I turned down the job. I went and became a lab rat in St. Louis. My postdoc mentor is now a member of the National Academy of Sciences. He has salmonella vaccines on the market that were in development when I was in the lab. And so in talking with a colleague about how to frame this talk, he gave me this quote, which I found really interesting. And he says it's a quote that people in the gym use a lot, bodybuilders. And basically it's that. Success only comes before work in the dictionary. So you can get the credentials and get the degrees. We have the system set up that way. And we turn you loose, and science is a natural selection system. Can you play at the highest level? What do you need to do to play at the highest level? You have to do the work first. And so I go do this postdoc. I was very strategic about it. I wrote a supplement on his NIH grant, paid myself a little bit more than the rest of the postdocs. I go to St. Louis, most racist place I've ever lived, by the way. My postdoc mentor and his research scientist, who was his wife, had 17 postdocs, 5 technicians, and 2 students in the entire wing of the Mallinckrodt building. And it was competitive, to say the least. And so the point is to learn what you need to learn and then go out on your own. So I did. So I go to the University of Georgia. I go back there because they had the research resources I needed. And the research resources I needed were those in which I could do agricultural research. I had already decided I didn't want to become a human pathogen researcher. I really wanted to work on stuff that other people didn't work on. And what was really cool about UGA, and I ended up in the Department of Poultry Medicine, which had the entire spectrum of translational research, from genes and proteins to implementation. And I was able to do the entire spectrum of those activities in my career. And I'll show you how that worked in the next couple of sections. How am I doing on time? Because I can talk. So again, here's what the work looks like, to give you an example. So you can Google search, Google and all these databases are really cool because all these came in my lifetime. You actually had to go to the library to find stuff when I was a postdoc. And one of the first things that I wanted to do when I had my own lab was to study those enzymes. And pastoral multocida, like I said, could grow really fast in blood. It's doubling rate. So bacteria grow by growing long and dividing into 2 cells. So it's logarithmic, 2 becomes 4, becomes 6, becomes 8. And the rate in which they grow long and divide determines that growth rate. And so their growth rate in blood is probably twice what it is in any sort of rich artificial media that we can make in the lab that we commonly use for stuff. It's quite stunning. To my knowledge, there is no other bacteria that can do that. And the only explanation to me is that this organism somehow nutritionally could access nutrients that are in blood, and it will have to do it with secreted enzymes. The only secreted enzyme it made was the sialidase activity. And so one of the first 2 papers that I published after I got my own lab was to create a genetic system for P. multocida. So we identified vectors that we could move into the organism, methods to move them that were doable, vectors in which you can clone genes and express genes, and then other systems to knock out genes. So I had the 2 things I needed now in order to go after this enzyme. And so you'll see this publication. Me, a postdoc's the first author, my technician, Adam, who didn't even have a bachelor's degree at the time. I hired him. He was working in somebody else's lab just part-time, and I needed a good tech, and I figured I could train him, and I did. And another technician, a master's student. So this work was done by 2 master's students, a tech with without a bachelor's degree and a postdoc. I find out after we did this work, 4 labs in the world failed at cloning this enzyme. And I'll tell you why. Because coming into it then with a different approach allowed me to be successful in this. And how did it work? Well, number one, I'm showing you a little bit of the protein sequence. So this enzyme had motifs that made it a sialidase. But if you look a little bit at the structure, right here, all these things right here, I think I'm looking at the right one, are knots that are in the protein. There were 2 reasons people couldn't clone it. Number one, those knots in the protein were toxic to E. coli. So everybody took the easy way in cloning. I took a different direction because I knew I wanted to move the clone back into the organism. So it allowed me to do what they weren't able to do with kits. So as a boomer, I will say to you, kits are fine when they work, but don't expect them to work right away because they will prevent you from doing the hard stuff that your competitors will fail at because you chose to take a different route. The other thing about the protein, which is really a little fascinating. So you saw Manu showed all these gels. And so, wait, go back. There we go. And so here's what the gel looks like when you run the protein. This was a purification of the clone. Well, all these knots in the protein prevented it from migrating in the gel. So you pour these gels on these little flat glass plates, you cut a slot, you put the prep in it, you apply an electrical current to move the proteins through by size. They separate out by size. That's what these bands were. This thing wouldn't migrate into the gel. All it would do is sit up in the well. So we start looking at protein structure and realize that these knots are preventing it from unfolding and migrating to the gel. Well, the way you make other proteins do is you just boil them. This one had too many knots to boil. We end up having to autoclave it. And for those of you in the audience that don't know what that is, that's a pressure cooker. So if you pressure cooker it for 30 minutes and then load the gel, it will migrate into the gel. So those 2 steps alone prevented these other labs, one of which was the world's expert in bacterial sialidases. And so I happened to be having beer with a friend who was having beer with another friend, and I didn't know she was out of that lab. And she emails me, Aaron, Margie Lee's got it cloned. And I get an email from the world's expert on bacterial sialidases as an assistant professor. And I'm like, oh boy. And he's like, you cloned it. How did you do that? And there it is. So it migrates in the gel. The cool thing about it is when we start looking, so here are the clones, and you'll see that there are 2. We found out that there were 2 of these enzymes in this organism by knocking one out. And so I had to be able to make mutants. I went to that postdoc. Now I can do the things that other people cannot do with this organism. I knock it out, it still has activity. We clone it back in. So now we got to go back and clone the other one. Now, you see every time you see a little bump here, those are knots. We had to autoclave that one for an hour to get it into the gel. So the paper comes out, we show that with the enzymes in E. coli, it can grow in solutions that have this carbohydrate, this sugar that's attached to protein. And this is how the organism grows so fast in blood. And so we made knockouts, we put it back in rabbits, we showed that it couldn't kill rabbits, it doesn't grow as well. But meanwhile, while this happens, somebody walks in my lab and says, I want to work with you. And she said, I want to be your postdoc. And I'm like, why? She says, because I'm really interested in Pasteurella multocida and we need diagnostic tests for rabbits. And I'm like, okay, why? And I'm a veterinarian. I know people love rabbits, but they're like, what, 14 rabbits as pets? Okay, that's not really true, but you know what I mean? She says to me, there are 500,000 research rabbits in the country. There are multiple papers that show that rabbits just even colonize a pasteurella and not showing any symptoms of disease will have all kinds of aberrant results and all kinds of tests that they use rabbits for. And so we had tests for rabbits. And I'm like, okay, I've got these enzymes cloned that are on the outside. There are a few antigens on the outside. Maybe they're handy. So we ended up publishing that paper. And then another one where we took the antigen and created a diagnostic test for rabbits and what the response was in rabbits, and that it actually correlated with colonization even better than the test they were using. The company that's the main company that veterinarians and lab animal facilities send their serum to for testing, Antec threw away their tests. They send all their samples to where we run our tests now. And 2 patents on it, and then continuing education for veterinarians on how to use this test for diagnosing rabbits that are carrying it. And you can see here, we have this test running at the University of Georgia. This lab right here was essentially built by a veterinarian that only does exotics. And so he licensed our patent on the test and then runs it and offers it to veterinarians and lab animal facilities. And nearly all lab animal facilities send the rabbit serum for the rabbit colonies to here for the test. Our test on the enzyme nobody else could clone from an organism that no one had genetic systems for, all from that postdoc. So don't be afraid to do the hard work first. The second thing I'm going to show you real quick is this. I taught an NSF summer workshop, and my TA that summer was defending her master's degree. So I go to her master's thesis to support Michelle, and her work was a molecular project describing the microbiome of the earthworm. And I'm sitting there the whole time thinking, I don't care about earthworms. But I'm with this poultry group, and you know, we really need to know what's in the intestine of poultry. And so we started this project where essentially we took chicken poop and we sequenced it a lot. And then we described not only what was there, but the ecology and the successional events as birds develop. And this is broiler chickens. And what ended up coming out of that, which was really quite fascinating for disease control was where we thought, number one, that it was stable. What we found is right around here where the diversity of the microbiome drops was when it was really, what's the word I'm looking for, where birds were really sensitive to intestinal diseases. It changed the way the poultry industry handled disease prevention, but it also changed the way the poultry industry handled microbiome. And what happened out of that? We got about 20 papers, I guess over, I don't know, 15, 20 years on that. And ours wasn't the first lab to do it, but apparently we did it best. And this is the paper that everyone refers to, the one that I showed you previously on poultry microbiome. But what came out of that was this. The poultry industry adopted the findings so quickly, and then they knew exactly where to implement probiotics and other microbiome stabilizers, that in that period of time, the poultry industry was able to go almost antibiotic free. Tyson Foods, which is the largest poultry company in the world, Tyson Foods produces about 2 billion broiler chickens a year, 2 billion with a B. Only about, well, no, actually they may be close to 3 now. And there's about 8 billion broilers produced in the country. Tyson's number 1, totally antibiotic free, partly because of the results of what we found out about how the microbiome develops. Now, I didn't have any funding for that at the time. I actually wrote an industry grant. I got some internal funding from Hatch funds. We went on to start the work and then to collaborate with poultry companies doing some diagnostics. I developed a couple of microbiome assays for them to give them an idea of when things were changing. And then I gave a ton of talks on it. And I mean, I got to the point where I would say I refer to myself as the queen of chicken poop. Literally, where I've been the keynote speaker and the invited talk at international poultry production conferences everywhere, New Zealand, UK, Asia, Canada, wherever they grow poultry. This is probably my favorite. So I was a speaker at this one, which was in a skybox on the front straight of the F1 track in Monza, Italy. And so there were teams training that day. So I'm talking to him. We get through the conference. They had race cars and instructors for us to drive. It was awesome. And they paid me for that as an invited speaker. Don't be afraid to do the hard work. And so, you know, of the distribution of type of research that I've been able to do, I've really focused on doing things that I found interesting, which makes it hard sometimes because you have to get money for what you found interesting. And then trying to figure out if it tells me, shut up, if it's actually useful for something and which audience you need to present that information to. I'm very strategic in where I publish and very strategic in where I give talks. And so results of the hard work, you'll see here, that broiler chicken community, almost 900 refs, the microbiome, the way up there. I've done a lot of antibiotic resistance work that gets referenced a lot because we'll do it by molecular approach as well as by systems approach. And so the results of the hard work, lots of recognition of expertise. So one of the challenges you will have as a young scientist is to be sure to publish. You can have a nice website that says you're an expert and everything, but boomers still run a lot of this stuff. We go look for credentials by publication. Publish. And don't be afraid to do those hard experiments that your competitors will not do in order to establish your expertise and your dominance in a particular area. I'll skip over that. It's the normal full professor crap. The hard work gives you professional success, but don't forget your personal success. So I always have to put up a slide like this to show that I'm not just a complete nerd, although I am just a complete nerd, but I've got some cool hobbies. So success, my family, we meet in New Orleans for Christmas. My mom and my sister are here. My husband's here, who's also a bacteriologist. And my hobby is motorcycles. And this was me this year. racetrack. I took a road trip to Mexico. I think we were south of Mexico City in that shot. And then that's somewhere on top of a mountain there in Giles County on the other side of Blacksburg. So that's the end. Thank you.
All right, Dr. Platt.
Oh, boy.
So number one, thank you for that, Margie. That was the most enjoyable talk probably I've seen in the last.
The
few
boomer
months.
talk.
I love it. I have a few quick questions, I guess. So that number one, congratulations on the assays and the patents and the application, such translational and impact in the field. I guess one of my questions, that first paper where you had all those difficulties and the world expert on sialidases contacted you. It went to J Bacteriology, which is a good journal, but did
The
you,
enzymology section.
right? So did you think it could have gone to like a higher journal because the impact clearly has been huge? And what was the thought process? Because I know as early people we might be like, well, maybe it's not going to be all that big. So I'm curious
So,
how
you
would,
know, yeah, I don't know what I was doing. Okay, and I will say that. So being the first, I was the first woman in my department. I was the first African-American faculty member in my department. I was not the first African-American faculty member hired in my college, but the other two didn't get tenure, so I was the first one tenured. First African-American in vet school. So it was a series of firsts, number one. Number two, I did not have a, I would say, familiarity with the support system that could have given me good advice on that. So perhaps it could have done better. I was so impressed that we could get it in JBAC. I mean, that JBAC probably was the hardest American Society for Microbiology journal to publish in the time. And the enzymology section is just biochemists. So possibly, and I would say that would possibly be true for a couple of other things that we published. I probably could have shot higher, but I didn't know enough about how the system to work, being the first to even contemplate it.
yeah. And then just my other question is a bit of a scientific nerd question. So I've never seen people actually autoclave proteins to get them into solutions. So I'm wondering if when you all were doing that, had anyone tried that as a process before? Or was that you all thinking outside of the box saying, we got to get this?
I live outside the box.
Yeah. I love it.
I mean, just,
Any more questions? I think we have time for one more. Yeah, go ahead.
Hello, thank you, Dr. Margie Lee. Your talk was very awesome. Hello, everyone. My name is Jatia Mills. My question is in reference to being specific or strategic to which journals you publish in. If you can just go a little deeper in that and how you decided to choose which journals would be more successful for you.
yeah. Oh, good. The mic's still on. It's tough, especially if you're doing translational research, because there's some areas that you're translating to that people aren't going to see the publication if it's in these other journals. And so in some cases, what we would do is we would publish the basic science stuff in a journal of our peers, so to say. So I go to the American Society for Microbiology journals, PLOS, that kind of thing, for those types of work. But where it's applicable to a particular area from the translational standpoint, sometimes I'll do a second paper. Like we'll run those experiments in parallel on the side, and then that data set becomes a separate publication in the journals that the people will read them and need to see it. And I use as a good example. So we did a study looking at microbiome and embryonating chicks in the egg, you know, because chickens are kind of the in-between evolutionary thing in reproduction, where mammals, the embryo develops like in a host, so to say, and in insects, they lay eggs. Well, chickens are vertebrates that lay eggs. So the question is, where do they get their microbiome, right? And so we hypothesized that there had to have been something that was transmitted down the oviduct into the egg, so baby chicks would hatch out with something. So we did some work with that. And then we looked to see if there were certain probiotic formulations that you could implant in the egg that would change the development of the microbiome. And so in doing that paper, I'm like, the people that need to see it are the guys who are developing products for use in the poultry industry. And if I put it in a microjournal, they're never going to see it. So I hit that happy medium. I put it in the American Journal of Veterinary Research, but then I posted a PDF in ResearchGate. So if you look at references to that paper, there's a dozen or so. It's no big deal. But in ResearchGate, there were almost 500 downloads. And then when I would go to a conference, the industry guys were like, I need to talk to you. I read that paper that you published about, putting this in eggs and what'd you find out here and what would be the timing and why wouldn't I do it at 12 days and that kind of thing. So sometimes it's hard. So if you're gonna have to choose, get enough data where you can split it.
Thank you very much.
But it's easier later in your career than earlier. So when you're building a career, you go for the best journals you can get. Later in the career, you can do what you want.
Thank you.
Thank you.
Thank you all.
Hi, everyone. My name is Roberta Lemos. I work with Dr. Warren Bickle, and I would like to invite everyone to join me in welcoming our last speaker this morning. Dr. Lance Collins was named the inaugural Vice President and Executive Director of the Virginia Tech Innovation Campus in 2020. Prior to this, Dr. Collins was Dean of Engineering at Cornell University in New York State. And as the first African-American dean at Cornell, Dr. Collins more than doubled the proportion of undergraduate students from underrepresented communities from 8% to 19%, and increased the enrollment of undergraduate women from 33% to 50%, while keeping graduation rates and average GPA equal among genders. Dr. Collins has received numerous awards and honors, and from those, I would like to highlight that in 2020, Cornell Engineering established the Lance R. Collins Fellowship. It was created to support engineering graduate students from traditionally underrepresented populations. Dr. Collins, thank you for accepting this invitation to share your journey from Westbury, New York to Alexandria, Virginia with us.
Thank you so much for the kind introduction, and just a couple of pieces of background information. So Dr. Monet Roberts up front here is someone who I met while at Cornell. She was putting together a podcast and she came to speak to the Dean of Engineering to see if he would be willing to contribute on the topic of I'm sort of forgetting imposter syndrome. And so, I was very happy to say, yeah, we all suffer from imposter syndrome from time to time, and it was a fun thing. The second thing is to the previous speaker that you're not the only baby boomer. I can assure you that. Oops. Okay, so let me do a little bit of history for myself here. So Westbury is a town in Long Island, and you can see from the arrow where that is. You see on the top left, I can figure out. The bottom. Okay, right. Those are my grandparents on my mother's side, grandparents on my father's side, my parents, and the question is, could you tell from this, where this guy was going? And, it was probably not so obvious. And so, there's been this kind of eternal question about nature versus nurture. So there I am again. And this young lady here, that's my daughter. And so, so what I can say about it is that I and she have, no obvious overlaps in terms of interest. She's very much a liberal arts person, and reads, a voracious reader, et cetera. I hated to read. I loved math. I was constantly solving puzzles as a kid. But the one thing I can say is that there is one feature that's fairly prominent in the two of us, and that we have some resemblance to a gerbil. So just so you know that she turned out better than than those early pictures. There she is in Ithaca. That's Taganic Falls behind there. So I grew up in Westbury, the Mighty Dragons, and I'm sure you all know that or recognize the dragons. And this is pretty hard to see. You could not find me unless you look carefully. So I was a little bit of a rebel even back then. I was a senior in this picture, which meant that I was supposed to get one of these fancy jerseys, but I just kept my gray shirt because I just liked it, liked being simple. So I ran track. That was probably my main sport, but I also played soccer. But I was a little bit of a nerd. So I was on the chess club. And that was, it was interesting to look at the fact that there were African Americans on it, but no women. And then the other thing that's even nerdier is the mathletes, which again is solving those puzzles. And here you can see not even a single girl on the team. So these were, very different times. So when I graduated, as I was getting towards the end of high school, I really didn't even know what an engineer was. And by the way, I should also mention, my area of interest is physical side of engineering, so it's not going to be medical as the previous speakers. So an interesting thing was that there was something called a minority introduction engineering. And this was a 1-week program where you could spend on a campus. I went to Lafayette College, and it exposed you to engineering in sort of large quotation marks. The thing that I fell in love with in that week was a game called, so you got to understand the timing of this, 1976. So this is just after the lunar landings. And so there was a game called Lunar Lander, in which you were in charge of controlling a lander. It moved in one dimension. It just moved from top down. And you had fuel, and you were in a gravitational pull. So you had to use your fuel carefully. If you used it early, you'd use it up, and then you'd accelerate and crash. And if you waited too long and used it, then you would be going too fast. So when you tried to, use the fuel in the retro rocket, you would crash. And so there was this sort of delicate balance that you had to discover. And I was hooked. I said, okay, I guess I'm supposed to be an engineer. So I went to college at Princeton, and this was really a period of time when the Ivy League was really waking up to the whole notion of diversity. I mean, in some ways, these were early days as far as that's concerned. I worked with a wonderful advisor. What was great about Princeton is that everyone did a thesis, and I did my thesis with Dr. Prudhomme. And the topic of it was the flow of foams, which is the kind of thing that only I get sort of weepy about, but nevertheless, it was incredibly exciting to work with him. But it really was an interesting time as well. And so we were, there were 3 of us in the class, among the first Black students to get a degree in chemical engineering. And so there was this real sense of being at the frontier and in some ways defining how to move that frontier forward. And there were all kinds of stories that could be told about that, but just suffice it to say that they got some things right. I can't say that it was a miserable experience, but they also got some things wrong, and there was a certain amount of overcoming the things that went wrong. From there, I went on to graduate school, and I fell in love with research through that thesis experience. And what was interesting is that I discovered that there was this program at Bell Laboratories that supported PhD students. And you can see it's a really impressive set of numbers that over this period of time of about 30 years that it ran, roughly, it produced about several hundred PhDs in electrical engineering, computer science, physics, math. I was an unusual chemical engineer that got that degree. But what was really remarkable about that group, I'm proud to be part of that group because there was an incredible levels of success for those that they got their PhDs. They went on to become, some became some became founders, many became academic leaders, some became tech influencers. It was really an impressive group. What's also kind of a little bit terrifying about that is that's a pretty substantial fraction of the total over that period of time. I mean, we don't produce, even today, all that many Black PhDs in these disciplines. I think I saw a statistic that computer science today on an annual basis, it's on the order of 20 or 30 a year. So it really is, in some ways I want to say, I wish I could say it was a problem that we put behind us, but I would say that's not the case. It's still a problem. I went on to graduate school at the University of Pennsylvania and worked with a wonderful person who's no longer with us, but Stu Churchill was my advisor. Very interesting character. And in some ways, my career was very close to his. While I'm a chemical engineer by training, if you look at my appointments at Cornell and at Virginia Tech, I'm actually appointed in mechanical engineering. I sort of strayed over into that area. And he was just an incredibly colorful person on so many levels. He was very, very esteemed, renowned member of the National Academy of Engineering. But he was also sort of an odd person. His most famous paper is in a paper that was entitled "An Equation to Fit Almost Everything," and it was the thing that got him the most attention. He was often invited to talk about all of the different problems, that span everything you could imagine about science, that this one equation was able to fit. And so he was definitely someone who thought out of the box, and it was very interesting to work with someone like that, because he got us to explore. He forced us to think about the problems in ways that we wouldn't otherwise have. Then I postdoced at Los Alamos National Laboratory, and it was another colorful individual. So this is Frank Harlow. He's also no longer with us. Well-known, innovative physicist. Honestly, any one of these three topics here would be a career for most people. The idea that he was able to do three of them is pretty remarkable. But he was also a renowned artist. Those are his paintings behind him in the picture here. And he was the expert on Southwestern pottery. So if you found a shard of pottery on your property and you wanted to know what it was, you took it to this individual. He was really quite remarkable, but very much a Renaissance person, and very much, again, someone that got us to think outside of the box. So my academic career went along this line. My entire career has been at 3 land-grant schools. It was not intentional, but it was just kind of interesting the way that worked out. So I started at Penn State. I moved to Cornell and then Virginia Tech. And so I'm going to say, I have 2 or 3 slides that talk to my research, and you're looking at a cloud in case you're wondering. So so one of the things that I'm known for is work that I did in understanding what's known as the processing of clouds. And so you think processing of clouds, it looks so static, but if you, the question that we were asking is how do they evolve as quickly as they do? That was an open question in the literature a couple of decades ago. And as I was saying, they look so static, but if you do time-lapse photography, you realize that no, they're actually quite dynamic. And so really trying to understand what that dynamic process was critical, not so much for meteorology and weather prediction, which is done very empirically. But if you're trying to understand climate over decades and centuries, that empiricism doesn't work. And so one needed a more fundamental model to understand it. And clouds are pretty tough because on the scale of the cloud itself, it's on the order of kilometers. The turbulence, which is sort of mixing this whole thing up, is on the order of hundreds of meters. The drops themselves are sort of millimeter in size. And they rain out at millimeter in size. They form in the micron scale. And then, believe it or not, the space between the molecules of air is actually playing an order 1 effect on this whole process. And so when you put all of that together, you're trying to understand a thing that spans 9 orders of magnitude. And in the physical sciences world, that's about the hardest thing you can do when things that are important are going on over many, many decades of scale. It's really difficult to figure out how to model something that's going on at the tiny scale, maintaining that as you're expanding it across the larger scale. And of course, we're interested in the globe, ultimately in terms of climate change, so you could imagine the challenges there. So it turns out no one individual could do all of that. So my work was using something called direct numerical simulation. So these are calculations that are done on huge computers, literally 64 billion grid points, just to give you a sense of the scale of it. And roughly that number of droplets were being tracked. But what was great about going to Cornell was that there were really adventurous experimentalists as well. So we were able to bring collaborators onto the project. So how do you experimentally measure something like that? Well, if you could create a pressure vessel, you can, in some sense, shrink all of those orders of magnitude of scale to into a physical vessel that you can put inside of your laboratory, which is represented there. Or you have a balloon up in the air that measures things in the atmosphere, which is doing field measurements. And this is just someone at Michigan Tech that was doing things, creating a cloud chamber with thermodynamics built into it. So essentially, this thing evolved into really an international program across multiple places in Europe and in Asia, because the problem's pretty important. And the reason it's important is that clouds represent the single largest source of error in climate prediction. And in particular, what's called the indirect effect. So human activity puts particles into the air that change the dynamics of the clouds. And it's that part of it that leads to the largest error. So we've been, in essence, studying that particular process. So here's an example of one of our simulations. And so the thing that was most recognized, I guess, in this work is that if you look, the particles the blue are the droplets. And they initially were uniformly distributed. And one thinks of turbulence, and all of the yellow are just showing regions of vorticity in the turbulence. And generally, you think of turbulence as something that mixes, right? If you have coffee and you pour milk in it, and you stir it up, you expect that you're making things homogeneous. But what's interesting is because the droplets are dense, the turbulence actually demixes it. So they started out uniform, but the regions that are like these, like here, become like little tornadoes that sort of fling the droplets out. So the droplets end up concentrating in these interstitial regions. And that concentration leads to dramatically increase in the number of collisions that go on with the droplets, which causes the cloud to evolve fast. And so this work then exposed the mechanism, and then the other work is that people are doing is then trying to connect that to make sure that that is really what goes on in the atmosphere. After all, this was just a simulation, and we need to always be confirming everything that we see in the simulations and so forth. So then, so that's a very high level overview of my academic career. But then from there, I moved on to really having an interesting administrative career, completely unplanned. And it was something that just at Cornell, there were a set of circumstances that gave me this opportunity. And in 2010, I was named Dean of Engineering. And you can see the much darker hair at that point. There were, with any sort of change of career like this, you are going to have opportunities that come, and what it comes down to is the degree to which you're going to take advantage of those opportunities. The first one that came along when I started as dean in July of 2010 is that then Mayor Bloomberg, the mayor of New York at the time, announced that there would be an international competition to build a new campus in New York City that would be focused on growing the tech ecosystem in New York, and that it was essentially going to be a land grant. So the idea is that the city would provide land and $100 million, which sounds like a lot of money, but when you're building a full campus is not. And the idea is that some university would be selected to then build a campus to help grow that tech ecosystem. And Cornell was selected, and that was a year-long competition that ultimately ended up creating what you can see here, Cornell Tech. And that was just fortuitous. And what is really interesting about it is that the institution had already set that goal. So the moment that the competition was announced, there was not any hesitancy in the senior leadership of the university, all the way up to the board of trustees. Everyone was behind it. And I was drafted in to write the proposal itself. And I worked with the Dean of Computing and Information Sciences at the time. His name is Dan Huttenlocker, and the two of us worked. And ultimately, after 1 year of competition, and the competitor was Stanford, so you can only imagine in the tech business just how competitive that was from a brand standpoint and everything else. It was knock down, drag them out. 1 year of my life that I'll never forget in some sense. But we were able to win, and it has transformed the institution. And it was really just recognizing there's an opportunity here, but it completely changed the culture of the institution, which was wonderful, not just in New York, but it actually served as a point of change in Ithaca, New York as well. Became much more entrepreneurial. Everyone became very interested in it, and you sort of built a reputation for producing entrepreneurs that was really wonderful. So as was already mentioned, we did the other experiment that was successful was growing the fraction of women, and I just have one tiny little anecdote about that. So the situation was that we were in the low 30% when I started as dean, and by the time I left, it was 50-50. So 50-50 is interesting because now you match the population. And the question is, and very few engineering schools had that bragging right. And so the question is, what did we learn in the course of that process? So every engineering dean in the country worried endlessly about how to grow the numbers of underrepresented minorities and women. It's just one of the topics that was standing in every dean's meeting that we had. And there are just all of these, there's a kind of common wisdom out there in terms of women, which is that women are drawn to projects of a certain type that have a sort of societal benefit, because that is in some sense intrinsic to the makeup of women. And so if you wanted to be attractive at recruiting them, then you needed to build a portfolio of projects that would have that element. What happens at 50% is that you realize that that's not true. That in fact, men and women are drawn to different kinds of projects in roughly equal numbers. There was no particular bias. That it really, that bias was reflecting the fact that there was underrepresentation. And in an environment in which there's underrepresentation, you're drawn to anything that's going to be welcoming, and a project that has a sort of societal benefit ends up attracting people that are more welcoming. And so that was the origin of that bias, as opposed to an intrinsic interest bias. And so that's what we don't know what the world would look like if we had the same outcome in terms of underrepresented minorities. The world will look different, but in ways that some ways we can anticipate, but we can't anticipate all of those. And just it's why we have to keep working, because we're a better self when we get there. And there's no question in terms of the health between male and female in a perfectly balanced world, there's no question that there's a greater sense of health there. And someday I joked with my successor that I hope that he would get to celebrate the point at which we balanced, the representation with society, to see what that experiment would look like. I didn't quite get there. And so I'll just, the other things you can see. So here I am. Now, this is a situation was that I had completed my 2 terms as dean. I was about to step back onto the faculty when I discovered that Virginia Tech was looking for a leader of its innovation campus. And you've already heard the story about Cornell Tech. So I was both, they were interested in me and I was interested in them because I'm one of, there are only a few of us that have been through the experience of both developing, designing, and actually building out one of these campuses. There's a lot of parallels between the 2 campuses. So one is that Ithaca, New York to New York City is about a 4.5-hour drive, which is almost identical to Blacksburg to Alexandria. Like they're almost identical distances. The rural sides are almost about the same size in terms of town. The urban side's not quite the same. New York's a little bit denser, but similar. And so there are lots of commonality as far as that's concerned. The other thing that's interesting is that President Sands at Virginia Tech had announced in his inauguration the notion of growing the presence of Virginia Tech in Northern Virginia. It was already an idea that was out there. It was waiting for the financing. And in some sense, there's a complete analogy to when the Bloomberg administration said, hey, we want to have this competition. It provided the land and the financing that we otherwise would not have. So very much in the same way, Virginia Tech jumped all over that opportunity. This is in 2017, and it originated with Amazon saying that they wanted to have their new headquarters somewhere out there, and 250 municipalities across the country competed. And ultimately, the Virginia Economic Development Partnership won that competition because it said, look, we're not just going to invest in tax breaks and making your financial balance sheet stronger. They don't need that, actually. What they need are technology and talent. And you hear that over and over again. And so they saw the investment in higher ed as more important than the investment in their balance sheets. And that's what ultimately led them to come. So here's the vision. The Virginia Tech Innovation Campus is both a place and a culture that unlocks the power of diverse people and ideas to solve the world's most pressing problems through technology. So there's a lot there. The place in a culture was very important to me. I learned that through the Cornell Tech experience, that it's about building a culture that's different than traditional academia, which tends to be a little bit of the ivory tower. It's purposeful and it's the right thing to do on a traditional campus. But in an innovation campus, we want it to be much more integrated with the broader industry and tech ecosystem. So here's the place. I think it's a beautiful building. I mean, if you haven't seen these images, I encourage you to go online and take a look at them. We're opening in 2024. It's an 11-story building. It's in the North Potomac Yard of Alexandria, if you know that area. It's going to have state-of-the-art facilities, and there will be dedicated K through 12 space. We felt that was important to build in terms of the long-term goals of in terms of diversity. So a couple of reflections. How am I doing on time? Okay. So a couple of reflections first. And what I'll say is I view this as an accident of birth. I don't know, we can celebrate it however you wish, but the truth is a lot of things happen because I happen to come along at the very same time that the Ivy League schools were saying, hey, we want to open up. They had never recruited from my school prior. And so there was just this, this window opened up that allowed these things to happen. So I mentioned, as I said, I was among the first group of Black graduates from Princeton Chemical Engineering. I was actually the first Black PhD from the School of Engineering and Applied Sciences at Penn. So effectively the College of Engineering. I mean, it's almost a little scary to think that, that person could be standing in front of the room, that isn't somehow a historical figure, but that's the reality. And then I was the first Black department chair at Cornell, and then the first Black dean. So I wanted to say a little bit. I had been asked to speak earlier about the issues of privilege, because I always feel like, in some sense, what are the takeaways from all of this? And to me, this has always been the most complicated thing. It's a kind of subtle underground challenge that has marked all of my life. And it's really understanding, are you really inviting me or not? You know, what are the real rules of the road in terms of opportunities that are either there or not there? So what is privilege? It's a special right, advantage, or immunity granted or available only to a particular person or group. So we all know what that means. And I have this corollary that a person can only perceive its absence. In other words, if you're experiencing privilege, you don't really realize you're experiencing it. You just sort of feel like that's the way the world operates. It's only when you don't, when you don't have it that you realize, oh, there's a privilege that you can see others are getting that you're not getting, and you become cognizant of it. So, like I use it as an example, suppose you're in a grocery store or just a convenience store, and the customer purchases a soft drink. There's no element of privilege associated with that. But then I say, well, all right, the clerk is a woman, the customer is a Black man. Okay, so now you start to see there could be a tiny bit of tension in this. And it turns out that this Black man happens to be the starting linebacker on his college team. So now you have the sense of big, powerful, and it's 3 a.m. And all of a sudden, the whole contextual information changes completely, just in sort of just describing these different elements of the situation. And that's the confounding thing here is that, so I might find myself walking in, before I had gray hair, I might try to disarm. 3 a.m. it's like I might say, hey, as I'm walking in, to just make it really clear that, I want to disarm the situation. Gray hair, turns out you have to do less disarming. It's one of the benefits. They're mostly negative. So then navigating it. And so for me, it's always been about mentors and having trusted mentors. And I call it a team sport. That means that you've got different people for different aspects, and you seek guidance, and that guidance includes things like, are they really ready for me? The issue of, for example, becoming the first Black dean is deans have to raise money, a lot of money. And I mean, will the alumni be able to make donations to a Black dean? Like, is that going to work out or not? And so, those are questions that you just can't reason on your own. So you have to build this portfolio of mentors. And I love this. This was from a former colleague at Cornell. He's no longer there. He's actually at Morgan State. He's in Virginia now. And he asked this question. I don't know if the audience is going to appreciate this or may not. It's maybe too dated to my generation. But what do Herbie Hancock, John Coltrane, and Thelonious Monk have in common? Yes, so there's at least one person out there that knows that they're jazz musicians, okay. But they all played for Miles Davis. And I feel like and I give credit, that's Mike Spencer, a dear friend. And he did this in a similar similar venue, and it was for me very profound, because it really said, well, while they're all great, nobody touches Miles Davis, right? And so just being in the presence of someone that great really makes a difference. So I look at my mentors. So I mentioned I was among the first African-American chemical engineers. You are now meeting the first. Cato T. Lorenzen graduated 1 year ahead of me. Now, people that are in the biomedical engineering community, he needs no introduction. He's a member of everything you can be a member of. There's no 3-letter organization that he doesn't belong to. The only thing left is the Nobel Prize, and I just hope we get to see it, because that's it. There's nothing else that he can do. So Cato's been a phenomenal mentor. He's also, when he was a student, he never encountered a B. He was a phenomenal student, and it just set the bar. It's like, I felt like if he could do it, there were no excuses. I always love being in a situation where it's like, I can't really kind of schlep along here. This is like, if this person 1 year ahead of me is able to do it, I should be able to do it. And Cato looks like me, right? He's, this is someone, but here's my second mentor, Kent Fox. And so he was someone who was he was provost that appointed me as dean. And he also recognized strengths in me that I didn't even know I had. And he was the person that answered the question, will alumni be able to donate? And the answer was yes. And it was very helpful in making me successful. And then I also spent some time with the chairman of the board at Cornell, who turned out to be he was an engineer and a wonderful human being. And we, he taught me that, not only can I do this, but it turns out that I was able to do it well. So I guess the other takeaway from this is that you need mentors. You need them, but they don't always have to look like you, so that's really kind of the message there. And then, of course, the third bullet here that, you're at some point, you're going to become mentors, those that are younger in the audience, and just realize that this is a process. So I was mentored by some, and then I want to be a mentor to others. So thank you. I think I may have run a little bit over, but I will step aside as needed. Thanks.
Do we have questions?
Lance, that was amazing, Lance. And I don't know if this is working.
I think it's on.
Oh, it is on. Okay. I did not know Cato was the first. I did not know that at Princeton. And so I guess one of my questions is, I know women were allowed on Princeton somewhere around that time. I'm just curious how women being on campus with also the first Black students, what was the environment like? Did they work together? Or I'm just curious how that went.
That's a great question. So I was not among the first Black students. I was Black chemical engineer. So just to put, but the timing of women and the timing of the first Black students was similar. And it was generally, I think in sort of like late 60s, I think was when the first, or early 70s, depending upon which school, and then roughly the same time frame for the appearance of the first Black students on the campus. Now, your point about did they work together is a great one. Yes, and so there was a lot of team activities, and in those days they were self-organized. So the teacher would say, "I'd like you guys to self-organize yourself into groups. You're going to work on a group project." Without exception, my group mates were the women in the class. And so the interesting thing is then you go to reunions, years later, and my white male classmates would say, "Why do the women they were all coming up and hugging you, and you've got like this deep relationship with them, and I barely remember them being in the classroom." And it's like, well, so it's sort of reflective of it. And I've seen this in, sort of in multiple venues, but there was the sense that those of us that were different could coalesce together, especially when you're forced to do these things on your own. So, we try not to, today I think we try to assign teams, et cetera, to avoid that very awkward feeling that of being the last picked.
So I've got a question. Is this thing on? Yeah. Okay. And you don't have to answer it now because you might be on the panel. But a lot of students, especially when they're hitting postdoc, assistant professor level, in diversity efforts in particular, are starting to get moved into situations that can give them leadership opportunities. In a lot of cases, I think it derails careers from the standpoint of building a science reputation. And so I spent a lot of years ducking it successfully until I didn't. And so when did you make that decision then to start adding that as a possibility? Because I think a lot of them are going to get it early. I got it early. And how do you maintain that balance?
So the very first serious administrative position I had was department chair. So I was chair of mechanical and aerospace engineering at Cornell, which I didn't include in that slide. And that was only a few years after I got to Cornell, and it was very much a surprise. I went to Cornell to be a faculty member. I had incredible opportunity to collaborate, so that everything was everything I was talking about with respect to the cloud was just taking off. And then suddenly I'm being asked to become department chair. And so all I can say is it's not for everybody. I don't think one should feel like this is a step in the ladder of all careers. It's really not. It's definitely a sidebar kind of activity. On the other hand, what I discovered is that there's this opportunity to make institutional change that you really can't do as a faculty, as a rank and file faculty member. And that was attractive to me because I could tell that Cornell was ready, that it was really looking to be different. So just as I was telling the story about the other Ivy League schools, Cornell is the only land grant Ivy League, and it was co-ed from the day it opened, and it had Black students on the campus from within a couple of years of opening. And its motto is any person, any study. The any person part of that was a statement in 1865. There was a thumb in the eye of, the whole Civil War and frankly, the South, saying, no, any person can come to this campus. And so it really was intentional. It didn't always live up to its intentions. And so I felt like I had an opportunity to help it live up to those intentions.
Thanks very much, Ken. That was a great talk. We've heard a couple different views of different types of backgrounds at school. So we heard about, for example, being at a university where there are more Black people, and one looks around and says, well, there's more people like me here. And then we've heard about some rather, I would say, almost classic white elite institutions and the role they've played. So I'm just curious, from your perspective, where you are today, where you've been. What if your career had put you in historically Black college situations and settings all through up to your leadership positions? How much of a difference might it have made? And what did it really mean to be, in many cases, a first or amongst a small group in these other, again, pretty elite institutions in terms of your training and experience?
So that's a great, almost impossible question to answer because you sort of only know the experiences that you had. I think it's important. So what I would say is, I do have to say, I look sometimes with envy when I see, the stories about, oh, look in Morehouse, and I've met a number of people over the years that have had that experience, because I feel like they were nurtured in an environment where. I think, your comment earlier about, you could show your full self, and I didn't ever feel like Princeton allowed me to show my full self. There certainly were elements that were kind of, off-putting, et cetera, that had to be overcome. So you built up a certain set of skills around setting your own destiny, setting your own North Star, in that kind of an environment that probably, in some ways served me well, in terms of, just a kind of stick-to-itiveness that was built up. But I can't say that I see that as the right outcome. I mean, all of these institutions are working hard to actually embrace diversity, equity, and inclusion, which is the right answer. The right answer is that everything needs to change into one in which we are all valued for both the things that we have in common and the things that make us different. And so I in no way want to sound like I'm endorsing the climate that I went through. It's not a good thing, but it did produce a certain set of skills that have served me well over the years.
Hi, thank you so much for a fantastic talk. My question is about whether or not there's a cutoff for the density of the droplets in your maps, where you have, the droplets move around, you have the turbulent flow, to where they actually will sort of like coagulate in a certain area. And the reason that I have that question is because our lab studies interstitial fluid flow, but in the context of tissues, and how movements of things like proteins can cause, like in formation of gradients with like convective flow, can cause cells to respond in different ways. And so could that be applied to our side as well?
Yeah, so the answer is yes. And there's been this work has sort of touched off a lot of different application areas, but I want to caution you on one thing. part of that clustering that was so dramatic in the cloud had to do with the fact that the drops are 1,000 times denser than the surrounding air. So that density difference really drives that degree of clustering to a very high level.
Okay.
You won't see anything at the levels that you saw in those images in the context of your application area, but you still see it. And so there has been a lot of work, around looking in biological systems, everything from just oceans to within the body type circumstances to see, can these same mechanisms play a role? And they do. It's just a little bit less dramatic than the one that you saw.
Okay, thank you so much.
Time for one more? Is it still on? Yeah. Okay. Wonderful talk. I mean, fantastic again. And my oldest son, his middle name is Davis because of a fascination with Miles Davis, among the greats. So I connected on that part for sure. Connected on many other things as well. But one thing you said that was kind of quick, but I kind of grabbed onto it, is that you said at the Innovation Campus, there's going to be K through 12 space because it's important for diversity. And then you kind of move forward. And I love that thought because I'm wondering, well, I'll tell you what my perspective on that might be, but I'll be interested in maybe the rationale there. To have diversity, you have to raise it up from the ground level. Would be my perspective. And it's from a land grant institution, that's our mandate, right? Virginia Tech, that is our mandate. It's not just to grow our medical campuses so that we can continue to recruit NIH dollars, though it's important. Sorry, Mike, because I'm a faculty here and this is what I'm doing. I'm writing Neuro 1 right now. But I would say if we want diversity, we have to raise it up. And I think that's embedded in there, but I'd love to hear more about what you thought and what your thoughts are on that.
So that's great. And you caught you were correct in terms of what, and I apologize. I was just trying to move through the talk.
No worries, but I grabbed that.
No, what I would say is that if at the moment, so I've set the goal to make the Innovation Campus the most diverse graduate tech program in the country. So I've set this very high bar. We're giving birth to the campus today. Let us not use the history of the last 100 or so years to set a lower bar. Like we should be at the forefront. Now, I can work to get there by hook or by crook in the short term by just in some sense recruiting or rearranging the deck chairs, if you like, as a bad analogy. But if we can out recruit other institutions in the region, then we can, but the long-term solution is really that there just has to be a more robust set of people coming into the technology disciplines. And that immediately gets you into the K through 12 space. You've got to think about how can we expose students. And so that longer term plan is that we're building K through 12 programming in. We have a director, and the goal is that we really are. So we're working with the Alexandria City Public Schools for obvious reasons, but we want to do it in a modular way, and we want to get computational thinking into their heads. And let's face it, this modern world, computers are going to be running more and more of our lives, and really just being a user is not the right outcome. You really need to understand what is going on behind the black box. And so our thinking is that we want to train that in the K through 12 system. Early on, it's just the thinking, and then eventually for those that are interested, maybe they're going to become the future programmers.
Awesome. Thank you very much.
Okay. Thank you all.
We are now going to take a short walk to Riverside 4. We'll have folks who will lead the way, so follow the crowd, and don't worry, you don't have to leave the air conditioning to get there. But if you're freezing just like me, just step outside for a minute and then come back in, please. During lunch, we'll have the opportunity to have some more in-depth conversations about Juneteenth, explore what it means in current society, and aspire to what we want it to mean for the future. There's some thought-provoking questions on each table that we encourage you to discuss and enjoy your lunch. And then we will meet back here at 12:30. Thank you.